Hello, my name is Joanna Schlosser. I'm a member of the team here at Accelerate Okanagan, and I'm going to be taking over for Jamie this afternoon. So welcome to the first afternoon session in the patient journey, which is making radiotherapy personal. I'd like to welcome to the stage moderator Dr. Deanna Taylor and Doctors Francois Bachon, Deidre Batchelor, Juanita Crook, Michelle Hiltz, Connie Herr, Andrew Jurassic, and Sepida Pakpour.
Shall the rest of the speakers join?
That sounds great.
Yeah.
Okay.
It's Friday afternoon.
Yeah, from the.
Yes. I'm there.
Okay.
So, good afternoon, everybody. So I'm the moderator. We're going to have each presenter present their information, and then we're going to have a Q&A at the end. We are starting a little bit late, as you know. We're going to buffer that a little bit during the Q&A, as well as we might run over maybe 5 minutes or so, but we'll make sure that we stick to time as best as possible, okay? So thank you very much. It's my pleasure to have Dr. Do I have make sure I say this correctly? Dr. Backert, is that correct?
Sure.
Bachon. Thank you. Thank you. I'm sorry very much. Can I get you to introduce yourself and then go ahead with your presentation?
Sure. I'm Francois Bachon. I'm a radiation oncologist in Kelowna, and I will co-present with Dr. Deidre Batchelor and Michelle Hiltz. So we're going to discuss quickly making brachytherapy personal. So I was seeing a new patient this morning, and then one of my new patients, who we offered brachytherapy to as main treatment, asked me if that was the technique that we in Kelowna invented recently. So as tempting as it was to say, yes, of course we invented brachytherapy, I don't think that would be a fair assessment. The truth is we probably made it better, but we definitely didn't invent it. So if you don't recognize those faces, it's because these people lived in the late 1800s, and they did most of the important discoveries about radioactivity and led to using radiation as treatment for diseases, cancer and other diseases. So Roentgen, and Becquerel and Marie Curie. And she had a husband, but she was probably the brain of that endeavor, so I just left her. And then their discoveries led to treating the first patient in 1901. And that patient had lupus, and they treated that patient using a radium tube against the skin. So to be fair, the first treatment with radiation was probably brachytherapy, not other types of radiation. So I think it's fair to say that Kelowna did not invent brachytherapy, as the first treatment was 1901, but I'll show you why we're trying to make it better. That was the hospital in Paris where it was done. So on the left, that was one of the applicators that was used for cervical brachytherapy many, many years ago. 120 years later, after invention of radiation brachytherapy, these are the applicators and techniques that we use today. So same concept, we still use brachytherapy as one of the most important part of treatment for certain cancers, but the techniques and the applicators that we use are quite a bit different than they used to be. When we say to try to make it personal, what we mean by this is try to put the patient back in the center of this idea. How we can do that? Well, instead of trying to find the patient for the right applicator, or try to just select the right applicator that we have in our cupboards for the patient that we currently treat, would be to create an applicator specifically for that one patient. So each patient, each anatomy is unique, so why not trying to create an applicator every time we have a new patient and try to deliver the best type of brachytherapy we can. Innovate in treatment delivery, which means, again, putting the patient in the middle of this, which means trying to make the treatment available, accessible, everywhere, closer to home, short, so it doesn't cost too much money, they don't have to miss too much work, and then they get the best treatment with the best outcome. And then one model doesn't fit all, which means each patient is different. So while using the same type of brachytherapy for all those patients, when we can modify it and adapt it to the unique patient. So in the next minutes or hour, you will hear a lot about this. So I think the way we do this in Kelowna to try to make it as much personal as possible is using 3 pillars. The patients, so the patients are our inspiration. They are the ones that force us to actually create better applicators and better techniques, and they inspire to research and development. So the research and development is what you'll hear the most in the next minutes. The next 2 talks are on technical development, and I think a bit later on there's more biological development and how we deliver brachytherapy, and training. So training of new doctors, training of graduate students in medical physics, and that we do that daily. So maybe I'll pass the microphone to Michelle for the next talk.
Hello, everyone. So I am going to talk about an example of what Dr. Bachchan was introducing to you. And this is called permanent breast seed implant brachytherapy. It's a type of brachytherapy for breast cancer. And if this apple represents a mastectomy, then the standard of care now looks like this. So it's called a lumpectomy, and they just remove the affected area of the breast rather than all the breast tissue. And you can see that the cosmetic outcome for this apple is much better than the mastectomy. And the trick, though, is that this has the same efficacy, so the same chances of surviving the breast cancer as a full mastectomy if it has radiation, if the patient has radiation as well as this lumpectomy disease. That doesn't look quite right. Okay, the slide's a little bit, a little bit funny, but on the right-hand side there, this is depicting that a standard radiation treatment after this removing of the breast lump takes between 3.5 and 6 weeks, where the woman has radiotherapy every single day. So it's quite arduous. And the reason that this permanent breast implant brachytherapy is so interesting is because it takes place over 1 or 2 hours in a single day. So the woman just comes for a single treatment, and then their breast cancer treatment is finished. So because it's a disease that can affect younger women who may have families that they need to care for, or mid-career, this is a really important feature. So what is this permanent breast implant? It's a procedure that takes place in the operating room. This is an image of Dr. Crook, who's here with us, one of our panelists, and Dr. Batchelor. And the treatment involves implanting permanently little radioactive seeds into the breast to treat it from the inside out. These seeds are about the size and shape of chocolate sprinkles, if you can imagine it like that. And they're implanted using needles through a template that guides the position based on a careful plan of where we want to put those seeds. It's also guided using ultrasound imaging. So this is an ultrasound image that shows the dark part there is like the cavity that remains when you take out the breast lump. And those bright spots are radioactive seeds that have been implanted in. So this works really well, but it's very challenging. And it takes the expertise, like significant expertise of our physician, Dr. Crook, to make this work. So as Francois did explain, we're doing a lot of research and development in these techniques to make them more personal and better for the patients. So I want to tell you quickly about one example of that. The details of this doesn't matter, but what we're developing is a 3D ultrasound system, and it can make a full volume ultrasound image, and then with this little blue part, the mechanically encoded arm, it can position that image with the template, so we know where the needles are in the image and we know where the seeds are. So I can give you an example of that. This is what a 3D image would look like. And then what it allows us to do is to say, if we want the needle to be in the blue spot, we image it and we can see the needles in the pink spot, so we know we've hit the right position. And essentially, this allows for us to be really personalizing this precision targeted treatment. So this is where we're moving with this. The other example this is a CT image which shows in green there, again, that region where the lump has been removed that we want to treat. And to do that, we always define a larger region, it's our target, to make sure we treat that area and the surrounding tissue. This is an ultrasound image of that same cavity. And you can see that they look pretty different, don't they? So if we could have this 3D ultrasound image that we could attach to the CT so we could see them both at the same time, then we might draw a different target here in red that encompasses, even better encompasses the disease. So this is another reason that we're invested in developing this specific tool to help with the breast cancer treatment. And I would say that this will enhance sort of personalized treatment goals for breast radiotherapy. So that's one example. And my colleague, Dr. Batchelor, will just tell you one more example around gynecological brachy.
Thank you. So gynecological cancers are cancers of the female reproductive tract. And although a lot of what the concepts that I'll present today can be applied to cervix or uterine or vulvar cancer, I'm going to focus on vaginal cancer. In particularly, post-hysterectomy, either recurrence of endometrial cancer or positive margins following surgery for endometrial cancer. So what you see here is a slice through a CT image of a patient. So a slice this way through a patient. These are the pelvic bones This is an applicator. So an applicator is something we use in brachytherapy to get the radioactive sources to the site that we want to treat. It can be any shape or form, but and still be called an applicator. In this case, it's a simple domed cylinder that is inserted into the vagina. These are the vaginal walls, this light gray stuff here, and this blue zone is our target. That's where we want to deliver radiation. So there are several, or a couple, of commercially available applicators for attempting to treat this site. This is the most common, which is what you see in our CT image. Oops, sorry. It is, like I said, a domed cylinder, and it has a single channel down the middle, which allows us to put a high activity radioactive source through that tube up near the patient's tumor. And with that, we can create a dose distribution like you see here. So this red line is our prescription dose. When we're trying to treat these cancers, we want to cover our target, the blue area, with prescription, but we don't want to over-irradiate normal vaginal mucosa. So we're limited in how big we can make our prescription dose volume because we don't want this area to get too hot. But because we've only got one channel, you only get a symmetric, cylindrical type of distribution. There's also an applicator available which has 6 equally spaced channels closer to the edge of a domed cylinder, like you can see here. And with that, you can make an asymmetrical dose distribution, but you still can't reach too far from the dome. The sources don't go right to the top, and you can't treat the top of the vagina. So what we do in these cases is, in addition to our standard cylinder, we add needles inserted into the vaginal wall. And currently right now, this relies entirely on the skill of physicians like Dr. Bachon, Dr. Crook, to get the needles where we need them to go. And they're doing this freehand and essentially blind. Like, they can feel where they want them to go, but there's nothing to guide the needles into the position or depth that we want them. So there's 2 problems with that. And one is we still can't get needles up here to the top. And the second is that if you do more than one insertion on a patient, they won't be the same. So to fix that, we thought, well, what if we know where we want the radiation to go in the end. What if we take that and design where we want needles and applicators to be? And then if we take that design and put it into a 3D printing software package, it can take where we've planned the radiation sources to be and convert that into a design for a cylinder with multiple channels through it for needles that can go out at multiple angles and can go up through the top. We can then print that and then provide an applicator which is designed specifically for whatever patient is in front of us. And this should allow us to deliver radiation both more accurately and more reproducibly. Oh, and we're back to the breast cancer. That was a mistake, sorry. Thank you.
Thank you very much. So, with the panelists that aren't, I know we're up here, we're down there, we're up. Would you like to, if you're not presenting, so you can actually see the slides? Shall we do that? Let's break form a little bit so that you're not craning your neck. And it is my pleasure to introduce Dr. Pakbor. Can you just give your brief intro?
Sure.
And then start your presentation.
Part of my presentation.
Perfect, thank you.
So let me just learn this one. Let's go back. This one goes forward. Okay. Good afternoon, everyone. I'm so glad to be here. I'm Sepide Pakbor. I'm a new faculty at the School of Engineering, UBC. And today, I'm going to talk about integration of the microbiome science into radiation, kind of personalized radiotherapy. It's a little bit weird, but I will walk you through and I'll tell you it does make sense. So first I'm going to give you a little bit of background about my lab, what we are doing in the lab, and a little bit of background about the microbiome science for those of you who may not be familiar with this topic. And then specifically I will focus on one project that I have in collaboration with BC Cancer and a lot of faculties on campus. And I will focus on that project, talk about its objectives, a little bit of experimental design, a little bit of touch base on the results, and the goal, the main goal of the final goal of the whole project. So I joined UBC in 2018. I founded the Biomedical Microbiome Research Laboratory, and with the aim of translating basic discoveries of the microbiome science into diagnostics and therapeutics. We are lucky to be part of the prestigious groups. I'm leading the Human Virome projects as part of the MIT Center for Microbiome Informatics and Therapeutics. I'm leading the microbiome science of the microbiome project of the Huffman program at Harvard, that we are working with nursing students to understand their microbiome and the childhood adversity on nursing students' productivity and performance. And also I'm an affiliated researcher with the Broad Institute, that is the prestigious group for cancer diagnostics, but mostly using sequencing. And I can say I'm very lucky to have a very strong and interdisciplinary group of students that are coming from different disciplines. We have students from engineering departments, from biology, molecular biology, to pharmacy and medicine. And this interdisciplinary group, all of them working together actually to answer these questions. But what is microbiome? What am I talking about? These are microorganisms. These are fungi, bacteria, archaea, viruses, and their genes, that they are everywhere. They are in dust samples, they are in the air, in the water you're drinking, and also inside and outside of your body. If I just focus on human microbiome, unfortunately for some of you, I would say that we are mostly microbes, not human. We are carrying 100 trillion of microorganisms in our body. They do a lot. They actually produce energy for us. They make vitamins. They regulate our immune systems. If you disrupt our microorganisms, we are disrupting our immune systems. They regulate our glucose level. And unlike some of us believe, and actually we think that microbes are bad, they protect us against pathogens. We need a balance of microorganisms in our body. If I weigh them, they are almost 1 kilogram. They're the same size as our brain. That's why we want to consider them as a human organ, and several factors affecting them, including treatment strategies. And one of those treatment strategies is actually radiation. When you radiate a patient, you disrupt the microorganisms in that habitat. It can be oral cavity, it can be vagina, it can be gut, and it can be colon. And by changing the microorganisms in that habitat, you actually can make the disease worse. So in my lab, we will look at the microorganisms. We want to understand them. We want to find out how environment and different disease, and how they impact each other. But as I mentioned, I like to look at how I can link them to applications. How can I use them? And the application can range from bioengineering to medicine, and personalized medicine is the perfect match for microorganisms. Why? Because when sequencing came in and we started looking at the human genomes, and just looking at what we were hoping that, okay, I'll go and find a SNP and I'll find a specific drug for that SNP in my genome. But unfortunately, the pharma couldn't catch up because we're dealing with a system biology. We are dealing with a system, we have a gap, and we don't have actual drugs for all these data we have based on human genomes. And there is no, I should say that we're almost identical in our genomes. But when it comes to our microorganisms, we're only similar in 10%. So we are very dissimilar, and by just bringing them into applications, we can actually personalize our treatments. Another thing I like to actually raise is that, yes, personalized medicine is interesting. We are all talking about them, but I think, and my colleagues also think that we need to change our perspective. We always look at patients as end users. Scientists, we do research, we pass it to practitioners and industry, we commercialize it, we pass it to end users. And if something happens, it should come back to us, and it may take 2 or 3 years to get into the applications again. If I get lucky with funding, with industry partners, everything, it takes time to get into applications. We need to change this workflow. We need to put patients in the middle, that they need to be connected to us, they need to be connected to practitioners, and we need to be connected. We make a sustainable loop to get to actually to solve problems. And without AI and without kind of cloud computing, it's impossible. And I think that we need to have this integrated approach. Having this integrated approach, we defined a project with several colleagues on campus and at BC Cancer by focusing at head and cancer patients. And what was our goal? We wanted to find actually digitalized and personalized stent for head and neck cancer patients. But why head and neck cancer patients? There are some statistics. There are 5%. It mostly happens in oral cavity, mostly happens in men. And there are different treatment modalities, but one of the major ones is the radiation therapy. And as radiation happens, several side effects occurs. And one of the interesting side effects is the overgrowth of fungi in the mouth. And these fungi start growing, and why it's growing, it's like your plant, it's like your garden. Just, you have all these plants in the garden, you go and pick them up. What is coming to grow? Not those plants, actually VD pathogenic plants come and grow. It's exactly the same thing. We're disrupting the oral habitat. These fungi, that's the regular actual commensal microorganisms that are in our oral cavity, it actually takes the opportunity, it starts growing, and it starts consuming the mucus in our mouth. And as a result, it makes it dry. As a result, it makes a very bad smell. If I return those microbes that they've been there before, or if I prevent overgrowth of these fungi, we can actually eliminate a lot of side effects. But I can say that it's been years that scientists are working with stent materials just to minimize the burning, because as you radiate, it will burn the kind of tissues. But these kind of stent materials, there is no standard procedures. There is no standard system. Some centers, they use these stent materials. Some centers, they don't use it. And it's funny because there's no systematic studies showing that these stents that have been used in the centers, they don't leach into saliva. They don't have any toxicity. And unfortunately, there is no microbiome studies. There are some studies showing that microbes will change. There is no fact, but we don't know if there is any biomarkers we can find at a specific part of the treatment, so we can change somehow our treatment. So maybe I add a probiotics, or if I add some kind of mouthwash to eliminate some of these side effects. So to fulfill these gaps, we've defined this project. It had 3 phases. It's a big project. We are at the phase 1. First of all, which material to use, and for who should we use which materials, and what is there any specific designs needs to be changed as the patient changes. The second one, we really need to check their effectiveness based on the personal differences of patients. And finally, we need to digitize it, which is going to be very, very hard. We are at the phase 1. The phase 1 itself has 3 specific questions. We wanted to find out if the properties of these materials changes as they expose to saliva and as they expose to radiation. Because when patients come, they give them a stent. These stents are actually, it's interesting, they buy it from Sport Check, I think. And they keep using it throughout the treatment. But it's funny, who knows if their performance changes as it gets used. The second objective, we wanted to know that the properties, the absorptions and the transmission properties. And the third one is the leaching properties. Do they leach into saliva? If they leach, can they impact the saliva? Can they impact their gut? So I don't have much time, but I can say that we are testing 10 different materials. And the first 2 is the current ones that have been used at BC Cancer here in Kelowna. The third one is the materials that of these stents. We wanted to know if the manufacturing affects their properties. And the last one is the material that we are synthesizing. We are embedding carbon nanotubes into the materials, and we are thinking with different concentration of carbon nanotubes, we can personalize our treatments. This stage is in situ. We are using artificial saliva. My students making saliva in the lab. So we don't use actual saliva, but the second phase is going to be on patients, hopefully. So just a brief kind of the results. Material changes as they get exposed to saliva. We are not dealing with the same stent as you're using it during your throughout your treatments. And also different materials have different kind of Young modulus. This is not the perfect graph, but on the x-axis you have the compressive modulus. On the x-axis we have different materials, and it just shows that the most materials and saliva exposures affect these mechanical properties. And this is absorption properties. Most of these kind of polymers, they almost have the same absorption properties, but the one that these three are our materials with the carbon nanotubes. And it's interestingly, as you increase the carbon nanotubes, you're improving the transmission. Less absorption happens. So if there is an implant placed in in the patient's mouth, maybe you need to change this material. Maybe different layers of materials needs to be used. And also at specific locations, we need to put some fillers that actually avoid any burns. The next step is going to test it, how the microbes, what we do, we are going to collect to recruit patients. We compare the healthy ones versus sick ones. I'm looking for biomarkers. I'm looking for microbes that can predict the side effects, or they can predict the kind of some kind of disease modalities. And also, if there is some microbes, we go and isolate them, we grow them, and hopefully we can use them for therapeutics. But not only microbes, we can look at their metabolites. This is the expertise of my lab. We can look at their metabolites. We can also look at their proteins. Maybe it's not the actual type of microbes, it's their metabolites that exist in the lab, in the mouth. And finally, how to digitize it. We need to use AI. We need to use, when the algorithms we're making in the lab, it would be great if we can put them in the knowledge base documents, and as patients come to your centers, they get informed about the whole process. And when you get to look at the specific biomarkers, we can communicate with the practitioners and help them to choose the best maybe materials for their design. And finally, they communicate with the industry, and they need to make that stent for them. And they know that, okay, what type of material to use, how many layers of them to use, and they are linked to practitioners, to UBC, to patients, and they also know which OEM to use to manufacturing it. I know it's going to be complicated. I know it's not something that we can do it in a year or two, but I hope with this work we can make the head and neck cancer therapy more personalized. Thank you.
Wonderful. Okay, so it's my pleasure now to ask both Dr. Durasic and Dr. Crook to come on up and do their presentation.
Good afternoon. It's not connected. Okay, so the changer for the
Just use the arrows here.
yes, I understand that, but how do I change the screen?
There we go.
Watch your red
Mystery
hand.
solved.
Big green arrow.
Yeah.
Okay, so Andrew and I were given the task of explaining to you in a very short period of time how we individualize prostate cancer radiotherapy through cooperation of brachytherapy and Raman spectroscopy. And this is going to be a crash course in both those things. I think you have a better understanding now what brachytherapy is than you may have before you came in this afternoon. But there's basically three ways to deliver radiation. External beam is the common type. That's what the head and neck patients get. It can be IMRT, 3D conformal, stereotactic, but they come in every day for several weeks and get the radiation beamed into the area that's involved. There's also systemic radiotherapy. Radiation can be given by an injectable isotope. Now this isn't used very often, but we use iodine-131 to treat thyroid cancer, radium to treat bone metastases, and then brachytherapy, which is internal radiotherapy. So To deliver this, you require an applicator. We've seen several to this afternoon already, which can be fitted on the surface or within a body cavity like the vagina, or interstitial needles which are placed into the target you need to treat. And then to deliver the radiation, we can use permanent low dose rate seed implantations, which are used in breast cancer that Dr. Hills was talking about, or temporary high dose rate, and that was used in the gynecologic cancers. Oops. Oops, okay, I went twice, just a second, back one. Okay. So brachytherapy, basic characteristics are that it treats from the inside out, allowing a very high radiation dose to be delivered. It's highly conformal, since the source placement is designed for the exact shape and size of the target. The radiation doesn't have to pass through normal tissues in order to get to the target because it's placed directly into it to start with, and then it falls off very quickly as an equation of 1 over the radius or the distance squared. So differences between LDR and HDR. LDR, the low dose rate, like the seed implantation, takes several weeks to months for the dose to be delivered from these permanently implanted radioactive sources. We use iodine for prostate, we typically use palladium for breast, and we calculate the strength and the number of the seeds required in order to give that required dose to the area we want to treat through the natural radioactive decay of that isotope. And you can see in the picture at the bottom, the seeds placed throughout the prostate. High dose rate uses a single source. It's the same size as those little seeds you just saw in the prostate, but it's about 10,000 times stronger than the permanently implanted radioactive seeds. We typically use the isotope iridium-192, and in order to deliver this into the patient, we need conduits distributed through the target through which the source can pass. And it will, as it goes through each of these conduits, whether it's a needle or an applicator, it pauses every 3 millimeters for brief periods to deliver the required radiation. And that could be a split second, or if you need to give more of that site, it could be up to 10 seconds. So it's very, very easy to precisely sculpt the dose. So high dose rate brachytherapy and prostate cancer are perfectly suited to each other. We have advanced imaging now which shows us exactly where the cancer is within the prostate. The high dose rate dose of radiation can be sculpted to deliver more dose to those sites of disease that we've seen on the imaging. And the delivery is extremely precise since the patient is anesthetized during the treatment and the prostate is completely immobilized because it's skewered with this scaffolding of needles. So the next series of pictures shows you how we do this. So you can see the needle insertion happening under anesthesia. The patient is anesthetized. He's lying there sort of, legs up in stirrups. And I'm watching on the screen, and you see the ultrasound image on the right side of the screen, and each of those green flashes is indicating the position of one of the needles that I've placed within the prostate. Once the needles are all in place, we need to track the exact location of that needle. We don't want to just know in 2 dimensions, X, Y coordinates, there it is. We have to know the whole length of the needle because that's the track over which that source is going to pass as it delivers the radiation to the target. From these images, we'll contour the prostate and any organs that are of concern, like the rectum, which is the brownish tube back there, the urethra, which is the yellow thing passing through the middle of the prostate. The prostate itself is in red, and you can see the needle is indicated in green. And then we're ready to do the planning. And this planning of the radiation treatment only takes about maybe 5-10 minutes. And in that, we are calculating exactly how that source is going to pass through each of those needles that's in the prostate and how long it's going to stay in each position in order to give the required dose. So you can see that we can sculpt the dose around the urethra, which is the yellow circle in the middle. We're completely avoiding the rectum, which is below. And we can tailor that dose exactly the way that we want through pre-designed constraints. Now we're ready to treat, so you can see transfer tubes connected up to each of those needles that's protruding from the prostate, and that source will go through each of those transfer tubes in turn into the needle, step through that needle. When it's done, it whips back into the machine, into the next transfer tube, and does that needle. We usually use about 16 needles, and the treatment can take anything from 15 to 20, 25 minutes. And the patient is comfortably asleep during the whole thing. So how does advanced imaging get worked into this procedure? Well, we have something now called multi-parametric MRI, which has been like a total game changer. For decades, we've looked at ultrasounds and CT scans of prostate. You can't see the tumor. Forget it. I mean, you know what's in there because the biopsy showed it was there, but you can't see it. Multi-parametric MRI will actually show you the site of the cancer within the prostate. And you can see here the top series is a T2 image, which gives us, you know, anatomic detail of the prostate. You can still see the cancer, but not as clearly. On the ADC map, which is a diffusion representation of the prostate, you can see the areas of high-grade cancer showing up like a big, scary black hole. And in this guy, we didn't even know he had that cancer beforehand because he'd had biopsies here from the back, and they showed a little bit of stuff here and there, but they completely missed that one. And so this gives us this really vital information. And then we need to transfer that information from the MRI to the ultrasound, because it's the ultrasound we're going to use for planning our treatment. And we just have to co-register the images and transfer where the cancer was onto our ultrasound images. And then when we come to do our procedure, we have another ultrasound identical to the one with that information. We know where the cancer is, which is that yellow thing there, and we set that to get a higher dose because that's what we really want to treat. So if the nominal dose of prostate is, say, 13 gray, this cancer is now getting 18 or 19 with each fraction of radiation because we know exactly where the money is, what we have to go for here. So talk about personalized medicine. I mean, it's amazing. So, HDR brachytherapy isn't new. We didn't invent it here. It's been around for over 20 years, and it's evolved over that period of time from giving multiple smaller fractions because people were afraid of the high dose rate. They were afraid of the power of that source, so they divided it up to be more like low dose rate. But we've gone gradually to fewer, smaller fractions, and the standard of care now is to do 2 fractions in separate procedures. And the 5-year results with that now are showing about a 3% failure rate at 5 years, so it's excellent, it's very effective. We have the capability to give the whole dose in 1 session, in 1 fraction, and we can do it safely, but it doesn't work. The failure rate at 5 years is over 20%. So we're going to come back to that. The other ways it's improved is when they had multiple smaller fractions, the men had to be hospitalized overnight with all these needles in place and then get 2 more treatments the next day. It was not very patient-friendly. We used to only combine it with external beam. Now we'll do it by itself. It used to be planned with a CT scan, which ended up taking the whole day. Now it's all done in 2 hours. So there's been a lot of improvements along the way. But let's come back to that single fraction thing. There was an intriguing article published just last year from Australia with just 5 patients. They were treated with 2 fractions of 10 gray, and they were biopsied before the first fraction and before the second fraction. And then they did genome-wide 3' RNA sequencing to see what the genetic changes were in the cancer as a result of that first fraction before you gave the second fraction, because something is fundamentally changing in that cancer. And what they found was quite amazing. There's strong upregulation of the p53 pathway, clustered genes for inflammation, programmed cell death, apoptosis, immune regulation are all massively upregulated by that first fraction. And all these things make the cancer more sensitive to subsequent radiation. So your first fraction is sort of put the cancer back on the ropes, and the second fraction is a knockout punch. And you cannot do it with 1 blow. So what we're trying to do right now, oops, there we go. In terms of, oh, you did that. Oops, this one. Yeah. So what we want to do back there is avoid administration in a single dose. Going back. Okay. This one is there. Okay, we did that. Sorry, this is confusing. We want to avoid administration in a single dose because we're losing all the advantages of the sensitization of the tumor. And what we're trying to do is to describe and quantify and understand those changes that are induced in the cancer by the first large HDR fraction that make it so much more sensitive to subsequent radiation. The upregulation or downregulation of genes, vascular changes, T-cell attraction stimulation, changes in cell cycle distribution. And with HDR, because each treatment is given under anesthesia, it's easy to get these biopsies at the time of treatment. The patient's asleep, they don't mind a couple of extra needles to take a little sample to analyze. So it's easy to get the material that Andrew needs, because one of the ways we're looking at and describing these changes is through a process called Raman spectroscopy. So now he's going to talk about that.
Okay. Yeah. Okay. Thank you. So my name's Andrew Drasic. I'm with the University of BC Okanagan in the physics department. I have to fully admit I'm not completely used to seeing a lot of anatomical pictures. I have none in my talk, so I take that as you may. So I'm going to change gears a little bit. Here we are. We've talked a lot about radiation therapy this afternoon, and I think you're getting a bit of an appreciation of what it looks like. So the patients will come in and they'll have some sort of imaging, planning, and so forth, and then the delivery. And we've talked about a couple different types of delivery: the external beam radiation, the brachytherapy, and so forth. In any case, there's some sort of an outcome. Okay, and that's what we spend our time thinking about. How can we monitor that outcome, or even have a bit of an idea beforehand what that outcome is going to look like. Okay, so if I were to sum up sort of in bullet points the objectives of our lab in a sort of a grand scheme of things, it's the goal we've set ourselves is, can we provide molecular level, spatially resolved, and I'll talk about what all that means in a second, information on radiation damage in cells and tissues, okay, with expressed idea for monitoring patient response radiation, or for developing of predictive assays, okay? And very loosely, what you can think about is we've got, you know, a bunch of data that we put into the problem, okay? And we're going to talk about one specific called Raman spectroscopy, which I'll explain in a second, and then some data analytics, which we'll clearly have to do to come up with some type of information which we can then make some sort of either predictive or monitoring-based decisions, and then have the oncologists act on that in some way, okay? So, what is Raman spectroscopy? Well, it's light scattering, okay? It's what we call, in the field, we call inelastic light scattering. Say you've got two molecules, okay? And you shine some light on those molecules, you're gonna make them vibrate, okay? If the molecules are different, say they're a different size, they're going to vibrate at different frequencies, right? This is where the quantum mechanics lecture comes in. What we're going to have is in real life, the frequencies are what we call they're quantized. They can only be very specific energies. So if we measure, if we shine light on our sample and then we measure the wavelengths that we get off of the sample, right? We spread them out like this. Pink Floyd, they were closet spectroscopists, so they did this. What we get is this thing which is called the Raman spectrum. Okay? And each bump in the Raman spectrum corresponds to a particular molecule or a particular class of molecules in the sample. That's the important piece here. Okay? So the way that we do it in the lab is through a microscope. Okay? So we shine our light through a bunch of stuff, which doesn't actually matter here. We shine it down onto our sample. And we're on an encoded stage, so that means we can get very precise spatial information about the samples that Juanita is giving us, about those samples. And we'll look at some of that information in a second. We shine the light, the scattered light comes back through our microscope onto a bunch of detectors, and we get this thing that's called a Raman spectrum. We can actually get what's called a Raman map, which we'll look at in a minute. But this is sort of the experimental way that we do it. The Raman spectrum itself is very, very complicated, okay? So there weren't quizzes at the end of some of these talks. There is for this one. No, but the important piece here is that what we've got is a Raman spectrum, and all of these bumps correspond to something, some kind of biomolecule in our sample. DNA, protein, lipid, cholesterol, da-da-da. So what we're getting is a very information-rich spectrum from our samples, and we're able to track multiple biocomponents. We've heard a lot about biocomponents. We're able to track multiple biocomponents as a function of radiation or so on. And we're able to do this all within one experiment. So this isn't like some of the gene sequencing stuff that we heard about where you're picking out very specific genes. We're looking at classes of molecules, but the advantage is we're looking at all of them at once, okay? So what can we do with this, right? Say we do a simple radiation experiment where we take some cells or tissues or whatever and we irradiate them, and we look at the Raman spectra as a function of time. So what's happened to those spectra as those cells have been irradiated. This is what the things might look like. So the blue here is an unirradiated spectrum. The red one is an irradiated spectrum after 3 days of radiation. We do some analytics, which are not important, but what we can actually do is pick out some specific biomolecules that are changing. I've shown you a picture here of glycogen. So we've actually talked a lot about glycolysis, glycogen and glucose. So the production of glycogen is something that we can very clearly see happen in some of our irradiated cells, okay? So for example, if I look here, what we've got on the y-axis is increasing glycogen. I've got 3 different kinds of cells, and I've got time post-radiation. So we're looking at the production of glycogen in certain samples and not in others. So you can already see how using this technique we can start to tell whether or not ultimately a patient, but in this case these cells, are responding to radiation or not. This plot here just shows us that when we look at the survival fraction of cells as a function of dose, we actually see that these ones are sensitive. So the green ones are sensitive. They didn't produce a lot of glycogen, so they just died. Whereas the blue and the red ones are more resistant to radiation, and they're trying to do something. They're trying to repair themselves by generating this glycogen or producing this glycogen, okay? We can actually radiosensitize the samples by using pharmaceuticals. So I'm giving you an example here of metformin, right? Actually it's an anti-diabetic drug, right? But what you see is this is our initial experiment where we've got the radio-resistant, right? And then when we add the metformin, we radiosensitize it, okay? It works for this cell line. It doesn't work for this cell line. So again, you're starting to see how we could use this technique to determine whether or not a drug is going to be useful in the treatment of cancer for radiosensitization purposes, okay? We've also talked a lot about the spatial heterogeneity of tumors. So I just want to talk about that for a couple of minutes. Here I'll just show you an example from a mouse model. So we've got this little mouse here, poor guy. He gets this tumor grown in him. We zap him, then we kill him, and we excise the tumor. We slice it up, and then we acquire what we call spatially resolved Raman spectra. So we acquire this data on a 2D map, okay? What we can see is that when we look at the production of glycogen, just think of white as more glycogen, black as less, right? We see that it's not uniform. We're getting this spatial distribution of production of these various biocomponents. Again, speaking to how we potentially use this technique in the aid of understanding how patients respond to radiation, because this isn't going to happen in the same way for different patients. We can do a lot of fancy math, which I'm not going to get into, to actually quantify how the heterogeneity of the tumor is changing as a function of dose and time post-radiation. Okay, so speaking a little bit to some of the importance of tumor heterogeneity. Okay? And finally, just I want to quickly show you one plot here. This is our first, hot off the press just a couple weeks ago, data from some of Juanita's samples, where we now are taking actual biopsies from the prostate patients that Juanita spoke about earlier. And again, we're able to see that the biocomponents are changing pre and post radiation. So we're showing 4 different patients here. Here's patient number 72, post radiation and pre-radiation, and we see this change in the biocomponent that we got simply by shining a light on the samples. Okay. So this is work in progress, but to us it's very exciting because it's showing us that we can actually port this not just from tissue cultures or mouse models, but to actual patient patient information. So this is very exciting to us. So in this sort of short whirlwind tour, hopefully I've kind of given you a bit of a sense that this type of work, this type of light scattering can identify changes in classes of biomolecules. It can be used to identify the efficacy of radiosensitizers, and we can actually port it to patient-specific biopsy samples. So in the future here, of course, the patient biopsy stuff is where we'll be focusing our work. Okay? And just as a summary slide here, this obviously doesn't happen in a vacuum. Some of our collaborators here, we encompass quite a wide range of expertise. We actually work within the framework of the Medical Physics and Data Analytics Cluster at UBC, which is providing financial support by way of their Eminence program. And we have data analytics experts, so they're in green. We have radiation physics or medical physics. We have radiation biology, and also radiation oncology colleagues. That's it for me.
Okay, so it's my pleasure now to introduce our last speaker before we go to the panel, Dr. Connie Hoer.
Thank you very much. Okay, so as you heard, my name is Connie, and oops, that was off. There we go. The big green button, not the red one. Okay, I'm going to talk about proton therapy, and I'm from Triumph, so I'm going to introduce Triumph very shortly, talk about ionizing radiation, different types, what we do at Triumph with proton therapy, the research that we're doing, and then I'm going to go on a tangent and talk about something somewhat related but different. Because Triumph is situated in Vancouver. It's Canada's Particle Accelerator Center. We are a bunch of physicists. I'm a physicist as well. We are on the UBC campus there, but we are owned and operated by 20 universities across the country from the West Coast to the East Coast. And it stands for Tri-University Mason Facility, and UBC is actually one of the founding universities of Triumph. So this is a map of Triumph. We have the world's largest cyclotron. We know all about this fundamental physics stuff, but all with the red circles, we're actually applying our knowledge to a medical application. Now, we at Triumph are very fortunate in the sense that we have access to a lot of ionizing radiation, not just to photons, which are typically used for cancer therapeutics, but also to ions. Here's an alpha particles, to neutrons, to protons. We have protons, we have heavier ions. And you can see when an alpha particle goes into matter, it goes in not very deep, and it has somewhat a dense ionization. All these circles are ionization sites. You can have electrons, a beta particle, that will go in and it's scattered quite a bit more. You have, of course, photons that scatter a lot, and there are a lot of ionization sites that are spread out. And we also have access to neutrons that are indirectly then producing recoils and protons that do all this. So why is this important? It kind of looks interesting, but it has a fundamental effect on if you're using which particle you use to treat your cancer. So if you use particles that don't have very dense ionizations, you have the top, for example, photons or electrons, sometimes protons. And when you go and you try to kill your cancer cell, and your goal is actually to try the DNA, and we heard in the previous talk that that's not the only way to kill a cancer, but it can be an efficient way to kill cancer. These low LET particles, they miss the DNA directly. So you're not even hitting the DNA. That's okay. Nature is actually very kind to us, and in most cases, that's just fine. You can use low LET particles, and the cancer cells die anyway. But there are certain types of cancer that are radiation resistant, and you need something that has a very dense track of ionization sites, for example, ions, neutrons, and protons that will destroy your DNA directly, hopefully with a double-strand break. The other result of this different behavior of the different particles is actually how the protons then go through matter. So here on the right, we have a plot the depth into tissue and then the dose that you give. And traditional radiation therapy with photons is the green line here. So you have a little bit of dose on the surface, and then it rises very, very fast. You have most of the destruction here, and then you have this real long exponential decay that basically doesn't stop. With protons, because they have a mass and they are quite heavy, they're 2,000 times heavier than the electrons that they normally interact with, you have a plateau coming in, so you have some dose on the surface, and then they stop all in this peak, and it's called the Bragg peak, and then they stop. And there is no dose afterwards. And the depth of the peak is only defined by the initial energy of the protons. So you can see how you can use this, of course, for treatment. You can, so most cancers is fine because your tumor would be about here, and you would actually treat with X-rays, and most of the destruction would be before the tumor, but you wouldn't go with the same direction each and every day. You would spread it out so your healthy tissue has a chance to recover, but you're always hitting the tumor. But for the radiation resistance, again, you can then nicely tailor it with the Bragg peak and hit the tumor with the high LAT area of the Bragg peak, and you have nothing afterwards. So you can have less dose to the surrounding tissue. You have lower dose on the incoming channel, no dose afterwards. You can spread this Bragg peak out. You can have very homogeneous tumor dose, and you can really, really easily control where it is just by the incoming energy. The disadvantages, you need a very high energy accelerator. You need a proton accelerator with 250 MeV. At Triumph, we have the world's largest cyclotron. It's 500 MeV, so just to get a scale for it, it's expensive. And therefore, it's not the standard way of treating. So at Triumph, we actually treated only ocular melanomas, choroidal melanomas. We started treating in 1995. We're one of the older centers. We actually ended our clinical operation this February. Our treatment was at the end of the lifetime, and more of these centers were popping up in the States, so we decided that it's more cost efficient not to refurbish our centers, but our patients are now being sent to the States. Here you have just a little bit when the protons are coming in from our main cyclotron. We always extract at a fixed energy, but the depth of the tumor is always different, so we need to change the energy, and that's basically just by dialing in the thickness of this field. More plastic means that you lose more energy in the field, so you make them more shallow. And then our Bragg peak is often too small for a tumor. A tumor can be much bigger than just a Bragg peak. For this, we have this modulator wheel. It's a wheel that spins. The proton will go through the side, and hopefully you can see this. It's very thin here, and then it steps up to thick, thin, and then to thick. So basically, you're creating a Bragg peak in the furthest depth, and then you're stepping your Bragg peak to more shallow and back forth. So you're painting basically your tumor this way in your patient. We have a library of these wheels in 1 millimeter increments spread, and they're not patient specific, but of course in the other two directions, horizontal and vertical, we have these custom-made collimators for the patient that then allow only the beam going through for the shape of the tumor. Here you can see a round one, here you can see a kidney one, so that's specific. So our treatment was actually very successful. I have to say, we only treated a quarter of the patients with the choroidal melanomas with proton therapy. 3/4 of the patients were treated with brachytherapy that we heard a lot about. And I personally am very proud of this, because many proton centers, they treat everything and everybody who comes to the door with protons. And I really believe that you have to choose your treatment to the specific patients. So we only treated the patients with the tumors that were too large to do homogeneous coverage with brachytherapy, or it was too close to the optic nerve or the optic lens, so you would have given too much radiation to those sensitive organs. So the 5-year local control rate was 97% for the optimized higher dose that we gave, and metastasis-free survival rate is an excellent 82%. And we got the quarter worst patients from that cohort of patients. So a little bit of research, what we're doing. So we talked about this beam shaping device, this wheel that shapes out the tumor. Here you can actually see. This would be in the flat tumor, that's the dose that you're giving to the tumor, that's the depth in the tumor. But this is the physical dose. We actually know that the biological dose is different. It's more like so, and this is ongoing research trying to figure out the exact shape of it. So we wanted to really make our beam conform as much as possible to the tumor shape, not with the physical dose, but with the tumor dose. So we looked into basically 3D printing these wheels, patient-specific, personalized, and then all of a sudden you can now have a physical dose that goes down like this, but you have a very flat biological dose. So that's one of the things that we did. The other thing is One of the biggest advantages of proton therapy is this Bragg peak, and there's no dose behind it, but it's also one of the biggest disadvantages of it, because nothing is behind it. So there's nothing that you can measure, nothing that you can really monitor during the treatment and making sure everything is fine. So you spend all this time on your treatment planning, all these hours, figuring out where to do the beam, and then you treat. And by the way, we only did 4 fractions. We always treated only 4 days in a row. And then you treat, and it's very anticlimactic, and you don't know if you actually hit the target quite well, and you go away, and after some months you come back and see if it worked or not. So we wanted to figure out how can we actually measure that we put the protons into the tumor or not into healthy tissue, because of these sharp fall-offs, it's more important than with traditional radiotherapy. And here you can see, in this example here, we have a brain. Here in purple is the tumor. So if you're hitting with your protons before, there is characteristic radiation that comes out. It's just nuclear processes, and you have some spectrum like so. Now, if we hit our tumor and we insert a specific metal marker into the tumor or very close to the tumor, that metal marker has a characteristic nuclear reaction and has a characteristic X-ray that you can measure. And we actually did a proof of principle experiment with this. Here is our facility. We have 2 detectors that measure these high energy X-rays in a plastic phantom that we put metal markers inside. And here, it's a little complicated, but this is the energy of the X-rays that we looked at. And we looked at moly markers, so that has specific nuclear reactions. And we can see that this peak, for example, only shows up if the protons have this energy, 14 MeV, but goes away if you go to 27 MeV. And this line similarly, and this line shows up at a higher energy but goes away in the lower energy. So we have really done a proof of principle experiment that with just metal markers, and in many types of cancer you have to insert metal markers anyway. If you choose the material correctly, you can now measure basically in real time without doing any computational exercises, do we actually hit the marker? And therefore, you can now really use the Bragg peak to its full advantage, because people, for example, for prostate cancer, when they use protons, they're so worried about that they're not hitting it, that they're actually throwing away the Bragg peak advantage. Instead of aiming the Bragg peak towards the sensitive organs, they come from the other direction. It's just fundamentally really weird to do that. So if you really know that you're hitting the mark, you can now really use the Bragg peak to its full advantage. And now just a side effect, a side note, this is pretty much a similar graph that we showed, so depth in tumor and dose. This is photons again, this is spread out proton peak. If you now go to heavier ions, for example, carbon beam, you actually get even more dose that you get into the tumor for the same entrance dose. There's no carbon facility in North America, but they are getting more and more popular in Europe and in Asia. And here's the map of all the facilities that we have in the world. It's very popular. We have been getting more and more. So we have 91 centers in operation, with more and more coming up and under construction. So just 2 slides on something that we're also working on at Triumph. Sometimes you have something like this. This is a prostate cancer patient, and obviously he has a lot of metastases all over the body. That patient had chemotherapy, radiation therapy, had hormone therapy, and everything failed. He had a life expectancy, I think, of just a couple of months. And therefore he qualified in Germany, where this case is from, for investigation therapy. And this picture, by the way, is from a PET scan. It's a gallium-68 PSMA scan that attaches nicely to the tumor, so you can then see the decay, and this is how you get a picture. Now, I'm not a chemist, so I don't want to insult anybody if there's a chemist in the audience, but if you, instead of putting the PET isotope on it, and you put a particle on it that sends out alpha particles, so 4 times heavier than a proton, so very high LET, it will accumulate in the same area, and that's what they did, and that's what happened. So that caused an immense stir in the field. Everybody now wanted to figure out, does this work? What else can be applied to? Is PSMA really the right pharmaceutical that you want to use? So there is an immense need for clinical trials, but there actually isn't enough of this isotope actinium-225 around. There's very, very low quantities of it. And we at Triumph, we are actually in really, really good fortune that we have one of our beamlines. So we have our 500 MeV protons, and when we're done with them, with the experiments, there's still a lot of them left, and we actually need to put them into a dump to get rid of them. The dump is right here. And we basically, just before the bump, we put our thorium target to produce the actinium, the isotope that can do this treatment. And it runs parasitically. It doesn't interrupt any of the experiments that we're doing. It runs 9 months a year. And we can actually make very cheaply, because everything runs anyway, more than enough to do all the clinical trials and a good fraction of the world supply when it goes into treatment. And thank you much for your attention.
I invite the presenters to come on up to the panel, to the front again. We'll do some Q&A. So first of all, just while everyone's getting set up, I just want to acknowledge the presenters and the audience for rolling with us with a rather dynamic start. I really appreciate it. You're trying to keep everybody on their toes after lunch. Just noting the time, I'm going to allow us to run a little bit over, but not much, so that we'll make sure you get out there for break. But the other thing that I wanted to say to all of you was just an incredible group of presentations, very, very complex information that I thought you presented very well. Key messages out of that I took were, talking about precision and a bigger bang for your buck. Also, that what I was thinking about when I was listening to you was what a wonderful example of a learning health system, where you were looking at the relationship between research, the medical, and the healthcare continuum. So thank you very much. We're going to turn it over to the audience now. I'm wondering if you have any questions or comments that you have, and if you want to just stand up, and if you need the mic, come on up. I'll hand it. If not, just speak up for the room so they can hear you.
Hello, everyone. My name is Deckard, and I'm here as a guest, and I'm very grateful to have this wave of information coming at me. I'm a breast cancer survivor, and I wrote my memoir as a patient experience piece of writing, so that all of the work that you're doing and all of your education, all of your research, and all of your experiments you're doing for me, the only thing I can give back to you is my experience from the other side. I chose to leave University of Waterloo and do the back to nature hippie log cabin lifestyle in order to avoid ever knowing what the word cancer even meant. And breast cancer arrived 4 years ago. To me, the word cancer just means you're gonna die, and the word radiation means even worse. So I learned a lot today, and thank you for everything you're doing, even though I don't understand a lot of it. And I've been very kindly given a table outside to make my books available to you. I would also like to do public speaking if there's any way that I can be of service to your students or research what it's like to be a patient. And I just wanted to show you how I managed to say yes to radiation treatment. Radiation coming through my left breast, which has only done good for the world. I've fed 4 babies for more than a year and a half each. When I was in this position, it reminded me of a falconer, and I decided to bring in my mind, I didn't learn the word visualization until later, my falcon to kill cancer cells. So that's how I was able to tolerate this very strange world that you're all willing to participate in.
Thank you very much for your comments. What's your first name?
Deckard, and I'm just outside, and
Okay.
I'll be at Chapters for book signings on Sunday in the afternoon.
Okay, so Eleanor, thank you for bringing your experience to us. You, of course, also remind me of another point, which is the great appreciation of all the patients that have participated in all of this research. So we're very, very grateful. Thank you for sharing.
Thank you very much.
Wonderful. Okay, is there any other questions out there? Oh, I'm sorry, Marilyn.
Good afternoon. My name's Marilyn. I am a patient partner in research. I'm a researcher. My question is on the brachytherapy. It's very interesting to me. I have a friend who has had this therapy. But my question is, how is it determined whether a patient gets just straight radiation? Someone very close to me, 44 radiation treatments for throat cancer versus the brachytherapy. Like, how is that determined? Is it the oncologist? They're just, how does that occur?
Would like to take that.
Here's the mic.
go on?
Yes.
It depends a lot on the tumor site. I mean, some sites lend themselves much better to brachytherapy, and you've heard about some of those today, gynecologic cancers, breast, prostate. Others, like lung or pancreas, esophagus, we use it much, much, much less frequently. So a lot is sort of predetermined by where the cancer is growing. If someone has a cancer that's in one of the treatable sites by brachytherapy, then it would depend on the presentation of the cancer. You know, for instance, with prostate cancer, if you can give brachytherapy, you can give a higher dose and you have a higher success rate in general. So we either give the brachytherapy by itself, or we combine it with some external beam, or we combine it with some anti-hormone treatment, or sometimes we do all three, but that depends on the presentation of the cancer. For other sites, like for breast, we don't tend to use it in the more advanced cancers because, you know, if it's more advanced and we have to treat lymph nodes anyway, there's no sense doing an implant in the breast if you've got to be treating the lymph nodes with external beam. So then you're going to treat everything all together using an external technique. So it's complicated. There's a lot that goes into it, but it shouldn't be just the physician's preference. It's based on the evidence, the availability of the techniques, and it's presented as an option to the patient, and ultimately the patient decides if they want to go for that kind of treatment.
Wonderful. Oh, good. Okay, another question. Go ahead, stand up.
I'll try
Sorry.
to-
You don't have to stand up.
Okay.
Apologies.
First of all, thank you. That was a beautiful panel. Really, I liked all the different angles. I don't have the background in radiation therapy either. But from the understanding of the protons, the molecules that are being projected to the patient, and then the experience patients are having, and all the just different lines of treatment. Thank you. That was wonderful. My question is directed to Andrew, actually. So I was curious. This is again a curiosity question. You mentioned biocomponents that you guys are measuring and kind of looking at all of them at once. Are there specific biocomponents that you guys are targeting? You mentioned glycogen. I'm just curious, are there some that you're really trying to measure as indicators of?
Yeah, that's a great question. As you can see, it's hard to pick them out, so that's one of the big challenges for us right now. And the way that we tackle the problem is we look in the first instance at classes. So the glycogen is a class of metabolite. Then we'd look for a generic protein signal and a generic lipid signal and that kind of thing. And then within the lipids, for example, we are able to break them down further into specific types of lipids. So it's really kind of a little bit of a hierarchical thing. I don't want to overstate it in the sense that it is pretty difficult. So it's actually something that we work very closely with our data analytic colleagues. What's the best way to pick these things out? Traditionally, in this type of work, you would end up picking one of those peaks and looking at that, but I don't think that's the way to do it in this case. So it becomes a little bit more complicated.
Wonderful. Thank you. Yes, go ahead.
Hi, everyone, and thank you very much for those talks, and thank you for the important and valuable work that you're doing. My name is Nicole. I'm from the Ministry of Health, but I would be really interested to hear a little bit about what personally, for you, what drives you to do the work that you're doing. And Dee, I'll let you figure out how to moderate that with that many people, but maybe a few people want to speak about that. I'd be really interested to hear.
Wonderful question. So why don't we, who would like to start? Because I think that's a wonderful question. Go ahead.
I can start with that. So I'm a physicist, and I don't work directly with patients. So I think for me in particular, that's a really interesting question. And I love being able to apply my technical expertise to helping the patients. I think that's ultimately the motivation. And one example of where that was really highlighted for me is in this permanent breast seed implant treatment, physics is involved when the patient comes and has a scan, and we determine from that scan whether they're eligible for the treatment, as this woman over here asked. And when I was with the patient, and I could tell she was quite nervous on this CT scan, and I said, I know, it's a big machine, it's noisy, but it'll be okay. She said, oh no, I'm not nervous about that. I'm nervous because I would really like to have this treatment, and I don't know if I'm going to be able to or not. And so I think sometimes those little personal stories that come forward to you, which keep you going. You know, because our ultimate goal is to make that treatment more accessible to more patients.
Wonderful, thank you very much. Does somebody else want to take? Wonderful, thank you.
Sure. I was trained in general radiation oncology at the Princess Margaret Hospital, and I was interested in doing a fellowship to enhance my skill set in a particular area. And I found out that you could get a fellowship and you could go and work in France. And I thought, wow, that would be really amazing to live in France for a year. So I went through the French medical literature to find out what they did that we weren't doing. And it was curative therapy, it was brachytherapy. I thought, wow, that sounds really interesting. I know almost nothing about it. Why don't we apply for that? And so I applied for this fellowship and managed to justify it for, you know, the learning techniques and bringing them back, etc. And to my amazement, I was successful. And once I was introduced to brachytherapy, it just made so much sense. I mean, why are we beaming in these huge, great beams and treating all this other stuff that just happens to innocently be in the way when you can put the radiation directly into the cancer and treat from the inside out? And furthermore, it's fun. It's a different skill set for every patient because you've got the challenge of placing these sources where they need to be. It's just, you know, you're never bored. It's a fabulous form of treatment, and applying it to as many different tumor sites as possible makes a lot of sense.
Wonderful. Does somebody else want to take?
Same for me, and I think to add a bit, and I'm sure Juanita will agree with me, is to do brachytherapy, it allows us to actually feel the cancer most of the time or see it, and then we can actually put the applicators within the tumor where we need it to be. And as we go on with treatment, in some malignancies like cervix cancer, we actually see the cancer getting smaller and smaller as we go on with the treatment, and we end up being able to tell the patient that we think at this point the cancer is gone. And there's something fun about the brachytherapy in a way that we can deliver the treatment to the cancer without treating everything else. And yesterday we were in the OR, and then we were managing to deliver 150% of what we wanted to give to the cancer that we could see on the MRI inside the prostate. So while treating the whole prostate, we were targeting where we saw the cancer with 150% of what we were planning to deliver. So it became so exciting during this procedure because we were putting needles at the right spot, and then we were able to tell the patient after that, well, actually, I'm pretty sure that's going to work because I wanted to give him 13.5, but I gave him 150% of what I wanted to give without giving him any more dose. So that's kind of part of brachytherapy, I think.
That's just a beautiful question. So thank you for asking that, Nicolette. And what was really clear too in all of your presentations, just how passionate this work is for you. We're going to have a couple minutes left before we go into break. But one of the questions I have is the obvious benefit for personal in terms of your passion in your work, the patient in terms of the treatment and the research in terms of science. I'm wondering if anybody would like to take a stab at answering. So what are the implications of this from a health systems perspective about this idea of being more precise in terms of bigger bang for your buck? Does anybody want to take a crack at that?
Well, as I was previously the president of the American Brachytherapy Society, so I sort of know both sides of the border and what the attitudes are. And one of my most difficult tasks being president of ABS was understanding why they had this reluctance and resistance to wide adaptation of brachytherapy. Because I said, well, it's so cost effective. I mean, we have a budget in order to treat patients in a catchment area, and if you can do that highly effectively and cheaper, like it's a no-brainer. Like what's the situation? Well, it's because it doesn't make money. You can't bill as much for a brachytherapy procedure as you bill for IMRT or SBRT. So that's why it wasn't catching on so quickly in the United States. But they're changing that, and they're trying to come up with a budget and a quota for this diagnosis. You get this much money, so treat it any way you want, and then they'll have to go for things that are actually more cost effective. But for a healthcare perspective, if you compare several weeks, 7 or 8 weeks of external beam treatments on the linear accelerator to coming in once for a procedure Sure, you have an outlay for the radioactive sources or the machine, the afterloader that we use, but it's still much, much cheaper overall for the healthcare system, and even more so for the patient, because they don't have all the travel costs or the accommodation costs and the lost work. I mean, it's definitely a huge, huge advantage for the patient. And if you're talking about personalized medicine, I mean, that's what we should be looking at it from.
Beautiful, yes.
I'm Heidi from Royal Inland Hospital Foundation and in Canada, so I'm just curious on that note, we have someone from the Ministry of Health here. So are there patients now who need brachytherapy but are not able to get it because there's a lack of seeds or because there's not enough product? So is that a current state that we are here in BC?
It's not so much a lack of the seeds or the product, but more of the time. Because if all the procedures for the whole catchment area are done in Kelowna, you know, we only have so much OR time per week. It's 2 days per week. We fill all our time, and we tend to treat people that are closer in our catchment area. You know, they get more priority than someone, say, coming from another part of BC. So it's just more our capacity to treat everybody, and it's the whole system. It's physics, it's the therapists that help us, it's everybody, because it all, you know, you all contribute. It's a team that does it. So it's not just a lack of seeds; it's a lack of everything.
Expertise and
Yeah.
Go ahead.
It's just important to know that the supply of radioactivity for brachytherapy procedures is completely different to the supply for other medical isotopes. So there's been in the news quite a bit in the past few years about shortages of isotopes for nuclear medicine, and that doesn't apply to brachytherapy at all.
And I think one of the challenges for the healthcare system is actually not that much if those techniques are available or not, is where they are available and the training and expertise in those techniques. So those techniques are very dependent on the user, and we're doing our best to train as many people as possible every year. Having said that, it is not available everywhere just because of the expertise. And we know that from data from other parts of the country or other countries that if the expertise is not available, then the availability of the treatment will go down. So patients will not have access to it. So that is one of the challenges at the moment.
Does anybody else on the panel want to speak to that? I'm going to wrap this up here pretty quick. Any final comments from the panel at all on that or any of the other questions that have come up? Any final questions from the audience? I think everybody, oh yes, go ahead.
I was just speaking up. So I was curious, like when, like when you implant of the radiation thing inside of the tumor tissue, during that penetration process, I mean, is there any possibility like the cancer cells come out of the tissue? I mean, before you penetrate the tumor, like the membrane is complete, but when you put the thing inside, like you made a hole out of the membrane. So it's possible that the cancer cells inside of the tissue go in the lung.
Yeah, that's not a bad question, and people have wondered about that a lot. The same phenomenon would apply for biopsying a tumor mass, because people have thought, well, if you stick a needle in my prostate, you can track the cells along the needle track and grow other little cancers. Well, it turns out for prostate cancer, it doesn't want to grow anywhere else. It's either going to be in prostate or in bone, basically. So they're not easily transplantable that way. And the other thing is that we're not, disrupting things as much as you might think. I mean, a biopsy actually kind of makes a hole and takes out a column of tissue, whereas we're sliding a needle in. We don't go right through and push the needle beyond where cells could be pushed ahead of the needle, say into the bladder or another structure. We stop at the edge, and then we're just bringing the needle back. And also you have, the radiation along the track of the needle as well. So it's a theoretical risk, but it's not one that is borne out clinically.
There are types of cancer that tend to spread along the biopsy needles, and there are specific types of cancer that we know about, and they have that behavior. Most cancers don't have capsules, so we don't disrupt the capsules. The cancers grow in organs without having a capsule around them, so that you implant a needle inside the cancer does not disrupt some kind of envelope that keeps it together. And as Dr. Crook said, most of the cancer cells have not acquired the ability to grow in other environments. So even though you would potentially displace them, if they're outside their environment where they're currently growing, they will not be comfortable enough to grow in other tissues. And that would explain why we don't see that happening in most of the cancers.
Excellent final question. So can you please all join me in thanking our presenters today? All right. Thank you very much.
Thanks, everybody. We'll be breaking till just after 3 here. So there's snacks and coffee and other things outside, so feel free to help yourself, and we'll see you back here at just after 3.
No, with like makeup on and like shooting videos. That was good. So I imagine that's what it means to me. But you want to do it anytime that you spend your thoughts
Welcome back, everybody. We're going to get started here with the last session. I'm going to invite Dr. Joan Batorf to come up and moderate, and the session is Empowering Patients with Choice.
Thank you. Thank you very much. Great to see that you're hanging in there. As this is the last session of the day, delighted you're able to join us. We have a great panel of speakers in this session on empowering patients with choice. You know, the journey through after a cancer diagnosis is a lengthy one, and that's a good thing because people are living longer after a diagnosis with cancer. But yet there's a lot of challenges along the way. And so our panel today is going to address some of the things that we need to think about to support cancer patients and some strategies to support people on that journey. So you're going to hear a lot of different ideas today, and I want you to keep track of your questions and comments, write them down, because after we hear from our speakers, there will be some time for questions and comments. So have them ready. We're going to begin this session with a presentation by Dr. Pippa Hawley. And I'm going to invite Pippa to come up and give that presentation. Thank you.
Thanks, John. Got sound. Okay, so I'm going to be talking to you about some work that I've done with the patients at BC Cancer and about their use on medical cannabis. And then Michelle is going to be talking to you about the basic sort of physiological aspects and pharmacology around cannabis. So I'm just going to start really with my presentation being to sort of set the scene, because from a patient perspective, I think the advent of access to cannabis has probably been the biggest game changer for them really in the last 15, 20 years. I should note that, sorry, I don't have a screen to look at here to remind me, so I'm going to have to keep turning around. But I am trying at the moment to get a study going where we're actually doing a randomized placebo-controlled interventional study on medical cannabis extracts for cancer-related symptoms. I've had like 3 hours sleep in the last 4 days because of Health Canada's ridiculous deadlines. So excuse me if I'm a little discombobulated. So this is just a little bit of the history of the medical authorization process that we've been through in Canada. And I think the best thing I can say about it is that it's better than nothing. That's about as good as it gets. Every time they've made a law that is supposed to achieve something, it's created another problem. And none of these laws have been satisfactory, and even the current laws that we have are not satisfactory. And this is one of the problems that's made it very, very difficult to do research, is because the legislative and the regulatory environment has basically prevented any research. Even now, with recreational legalization and delisting of cannabis as a narcotic, which is not a narcotic anymore, it's been a year, and it's still virtually impossible to get research through. Literally, it's been hundreds of hours with Health Canada and absurd deadlines and stupid questions. It's really almost deliberately sabotaging the process. So I don't want you to perceive when we talk about the lack of evidence for medical cannabis as evidence of lack of effect. It really is just lack of any evidence. And it's not because of lack of trying. So these are some of the countries around the world that do have access to medical cannabis. As you can see them all up on the end there. And there's a nice little map here which shows Uruguay, lone little Uruguay, now being joined by some of us in North America with an actual proper medical access system with its warts and wrinkles and its limitations, and also recreational legalization. There are some other countries like South Africa and Georgia where it's not illegal for recreational or medical use, but they have actually no system of access. They just don't put people in prison if they get caught with it. So this is a huge, really a big natural experiment, and we have no idea what's going to happen. There could be significant benefits, and there may well be significant harms. You may have heard some of the misreporting about road traffic accidents when they did the legalization for recreational purposes in Washington. They were forecasting carnage on the roads, and there was this report that was a 50% increase in road traffic fatalities. It was complete misreporting, which unfortunately plagues a lot of medical cannabis discussion. it was actually that there was a 50% increase in people testing positive for cannabinoids when they went into the hospital and had a road traffic accident. The actual fact was that there was no change in the number of actual accidents. In fact, it went, if anything, went down slightly. So there was no increase. It was just that they were actually testing, which they didn't do before. And this actually ended up being published in the BC Medical Journal, not peer-reviewed, and then taken on by the media, who went with the BC Medical Journal as being a reasonably reliable source of information. And a ton of really wrong stuff was published about it. So the survey that we decided to do with BC cancer patients, we picked 2 separate days. It was a couple of months before legalization, which was October last year, and 3 months afterwards. And we just picked random Wednesdays in a week that didn't have a public holiday. And we basically took everybody that had an appointment at BC Cancer for whatever reason that day. We had some that didn't have cancer at all. They were coming for hereditary cancer counselling. Some that had cancer and were cured. Some that thought they might have cancer. Maybe they had a positive FIT test and they attended for a colonoscopy. Could be anything, or it could be that they were having active treatment heading for a bone marrow transplant for lymphoma. It could be the whole 9 yards. We even had one who was a family member who actually got in because she had a cancer agency number, though she wasn't attending for her cancer at that time. And we had a mix of questions. Some of them were just tick the box, and usually at the end there was a space for people to write comments. And these are kind of questions that we asked people. We wanted to know what they were using, why they were using it, where they were getting it from, and what their challenges were or successes with access, and what their perception was with whether it was helpful to them or what problems they might have had. And this is the consort diagram. You can see how many were sent out. And I think the most remarkable thing about this is how 5 months apart we had almost exactly the same outcome in terms of numbers sent out, numbers returned. And in fact, the demographics were almost exactly the same as well. So I think that says something, even though the response rate was not a huge response rate, for a mail-out survey, 27% is actually pretty good. That's about as good as it gets. And the fact that the demographics and the numbers were so similar between the 2 surveys suggests to me that changes we saw between the 2 survey cohorts are probably real. So this is just a quick look at the demographics. You can see the age was, if you divide it up into people who've never used cannabis for any purpose, this is medical and recreational, so those first 2 columns. The number 1 and number 2 were the first cohort and the second cohort, so all those never users, the average age of 70, a slight preponderance of women, almost, well, not exclusively Caucasian, but vast majority Caucasian, and quite a high rate of high school graduation. And then if you go to the next 2 columns, you see people who used to use, most of them were using it recreationally, and then the last one is current users. And you can see that between the first and second cohorts, there's really not that much difference between them at all. Okay. And if we actually look at the proportion of patients who are using, the nevers here are never used it, not using it now. The middle ones, prior users, mostly recreational. And then you look at the current users, there was actually a statistically significant increase in the use of cannabis for symptom control. In the second cohort. So recreational legalization impacted medical use, even though there was no change in the medical access system between the first and the second cohorts. And look at the numbers, like nearly a third now, that's 29% of patients. That's actually a very significant proportion of current patients. And if you look at actually the ones that really had cancer and are currently having treatment, I think the numbers would be significantly higher than that. I don't know how many oncologists are in the room here, but most of them, when I ask them how many of their patients are taking cannabis that they are aware of, most of them say, oh, at least half. So it's a big deal. And in some situations, it could be a problem, especially if people are on immune therapies, as was discussed a little bit earlier, with the need for the immune system to be able to do the work. And if you suppress the immune system with an immunosuppressant like medical cannabis, especially with high CBD, that could be a problem, and we need to know about it. So looking at the routes that are used, the oils are the most popular. eating, creams, drinking, suppositories, these are all different options, but definitely oils are the most popular. So it's disappeared for some reason there, but the smoking is the second one there. So smoking has not disappeared, even though we tell people not to smoke and not to vaporize. People are still smoking and vaporizing because that's what they know. Most people actually aren't really aware that there's any alternative routes. So if they're going out looking for it, that's generally what people ask for. You have to actually inform them that there are oils as alternatives. And there were some changes between the first and second cohorts. Again, this is assuming that the use is the same amount, so this is percentage change, not absolute number change. And you can see that from before to after, we had increase in oils, increase in smoking, increase in eating, and a marked increase in suppositories. And I think that's a bit of a cultural thing. There was a sort of fad that went around that there was a misconception that people that take cannabis by suppository can't get high. So people who didn't want to get high were all going out and buying suppositories. And of course that's ludicrous because the THC is absorbed just as well from the rectum as it is orally or by inhalation. But it was a thing that went around, and it really made a difference to the whole province's use of cannabis. So looking at the current users, where did they get it from, and did they have an authorization? You can see with the second cohort, where a lot of them, there were new users. Those new users were certainly not getting it with the knowledge of their physicians. A lot of those patients did not have medical authorization, so over 2/3 of patients are using cannabis for medical purposes, but not using the medical cannabis access system. So the state of the recreational access system is really important for medical users, and one of the problems with it is that people who work in it are prohibited from giving people medical advice. So it's not really very helpful. Still many people getting product from friends, increased in the second cohort because a lot of the dispensaries closed down when they all had to apply for licenses. So their access got worse, and it pushed people into more illegal sources. Less people were going to dispensaries because there were less open dispensaries. The licensed producers were about the same. So people that had started with a licensed producer, most of them stayed with them. Homegrown didn't really change very much. You can see there was quite a big increase in the number of patients who were reporting difficulties with access. Again, this is probably because of the closing down of the dispensaries. So the reason why people like to go to dispensaries is they like to see somebody in person, and they want to buy things other than dry plant product. And the licensed producers online are only allowed to sell either dry plant product, or some of them are allowed to sell oils. Only about 1/4 to 1/3 of them do the oils, and they're significantly more expensive than the dry plant product. So this is why people prefer to go to somewhere where they can actually talk to somebody. The other problem is the amount. If you go to a licensed producer, you have a minimum amount that's kind of worth buying and getting shipped to you, and it's a lot of work to invest in time, invest in doing it. Whereas if you just go to a walk-in dispensary, you can buy a little bit of something, take it home and try it, and if you don't like it, you haven't wasted a lot of time and energy. I actually, just for the purposes of demonstration only, tried to sign up to see how long it would actually take, to the point where I was told I can now make an order. And I've been through 3 different licensed producers now, and I've started about 6 weeks ago, and not one of them have I managed to completely negotiate the problem. And this is somebody who's, I think, considerably computer literate, like I can do online forms, and I have a telephone, and I have an address, and I have a bank account and a Visa card, and I've still not been able to do it. Apparently one of the biggest uses now in public libraries for the staff there is for them to fill in medical applications for medical cannabis. That's what they're having to train the librarians in the public libraries to do now, because a lot of patients, they just cannot do it and they have to get help. So just whizzing on very quickly because I don't want to take too much more time, but looking at the reasons for use, pain, insomnia, anxiety, lack of appetite, they're probably the commonest ones. Tiredness is in there as well, but I think that might be mixed up a little bit with sleep disturbance, because many of our patients do take it for insomnia as well, and that can, I think, can probably be combined there. And before and after legalization, you can see there was a little bit of an increase in use for insomnia that was particularly noticeable, but most of it was pretty much the same. So these are just some comments I thought were worthwhile putting up there. You can read those. They were overwhelmingly positive. I know we only had the 27% of patients that were surveyed actually did respond, and maybe the people who didn't respond perhaps would have been more negative, but this was an opportunity for them to state their experience, both positive and negative. And the experiences were overwhelmingly positive, and patients felt very strongly that they should have access to cannabis for medical purposes. So in summary here, you can read it up there. I think you kind of get my gist really, that very few of our patients are accessing the proper system. And so it behooves us as care providers to be informed and try to help our patients so that if they do decide they'd like to pursue it, you at least guide them as to how to do it properly. Because if you don't, they're going to do it anyway. So think of it almost as a harm reduction. Even if you don't approve of their use of medical cannabis because you think it's not healthy for them for some particular reason, they're going to do it anyway. So just saying no or not engaging with them is not helpful. You have to be knowledgeable, and you have to explain to them how to do it and how to do it safely. Start low, go slow, keep the THC content down, and talk to your patients about how it's going. If someone tries it, try and only do it with one change so that you know whether it's helping or not, or harming or not. And then just like you would if somebody was starting on gabapentin or morphine, or an antiemetic, you try something, if it doesn't work, you stop it, you try something else. And medical cannabis is just the same as that. Okay, I think that's it for me now. Michelle.
Great, thank you very much. Okay, now I'd like to welcome one of our fabulous doctoral students from UBC Okanagan, who's doing her doctoral work in psychology, Michelle St. Pierre. Welcome.
Okay, so I think that it'll actually be a nice compliment to Pippa's talk. Thank you so much, Pippa. That was just really great information. As Joan said, I'm a PhD student in clinical psychology at UBC under the supervision of Dr. Zach Walsh. I've been studying cannabis for about 5 years, and my primary focus has been cannabis and mental health, and cannabis in the treatment of pain. So I do want to disclose that I have been paid as a graduate student research assistant in Zach's lab, and it was for a study looking at cannabis for post-traumatic stress disorder that was funded by industry, because there's unfortunately not a heck of a lot of funding from the government for this type of research. Okay, wow, yeah, so the font is big when you're this close. But cannabis has been used for pain for longer than written history. That was our medical texts, the very first ones document cannabis for the use of pain. And I wanted to draw your attention to this great article from the New York Times. They found a mummy, 2,500 years old, and it turned out that they think this mummy died of breast cancer, and she was actually buried with cannabis, and they think that she was using it for the treatment of that cannabis, or was at least consuming it for some reason. So humans really have an affinity for this plant. Cannabis is a broad kind of term. It's actually a bunch of different drugs or compounds. So the most sort of the star players are THC, so that's the one that everyone knows about, makes you high. And then there's CBD, which is really emerging on the scene right now. People are pretty excited about it, kind of touted as an anxiolytic, an anti-inflammatory. And then there's these things called terpenes, which are incredibly exciting, and really where I think the whole sort of science of cannabis is moving. They're what give cannabis their smell. So there's over 200 of them. We don't know what all of them do yet, but something like a lemon would have limonene in it, and cannabis also has limonene. Something like a tree may have pinene. Cannabis also has pinene at different levels, and these are actually shown to be they have therapeutic effect as well. So that might actually be where some of the therapeutic effects coming from. So kind of keep your eyes peeled for more research there. So we've been using cannabis for thousands of years. There must be a reason. And I think that that reason is likely because our body has sort of these keyholes for the keys that are the cannabinoids. So there's a graphic right there showing the endocannabinoid system. And it's touted as being a sort of homeostatic regulator. So this little nice poem here, relax, eat, sleep, forget, protect. That's what the endocannabinoid system does. It's the most numerous receptor system in the body. There's 10 times more cannabinoid receptors than there are opioid receptors. And the most important thing, I think, on this whole slide is that they do not occur in the brainstem. So in terms of the risk profile, cannabis is doing pretty good. You can't take so much that you have a respiratory depression, much like you may have with opioids. So before I get into what cannabis can do, I thought it was important to pull something from the National Academy of Science and Engineering and Medicine report that was released a couple years ago that did report that there was no statistical association between cannabis smoking and lung cancer. Certainly not up here today to say that smoking is safe in any way. I definitely think we should be encouraging all cannabis users away from inhaling any burning material. But I do think that that's important to just kind of put out there. And then, so now I'm turning to kind of the benefits of cannabis. Well, it seems like it would be a really great drug for someone who's going through cancer treatment. There are antiemetic effects, appetite stimulation, pain relief, improved sleep, decreased anxiety, improved mood. So a kind of a host of benefits. So to start, the antiemetic effects of cannabis. So I looked at a few different systematic reviews. I pulled some of them up there for you. And what they found by and large was that cannabinoids were moderately more effective than the widely prescribed antiemetics. But kind of what I thought was interesting from my perspective was that the patients preferred the quote-unquote beneficial side effects. So the euphoria. So they may be treating this one condition, but the side effects that they're also kind of are coming along with that treatment are actually something that they're seeking out. This next one probably isn't a surprise if you've been around cannabis culture much, the idea of the munchies. So patients were finding that after you go through chemotherapy, sometimes you can have a decrease in like food palatability. So food tasted better, appetite was increased, and they were actually eating more calorie dense, sort of fatty foods. So pain relief is something that I'm particularly interested in. When it comes to pain relief and cannabis, I think broadly what we can say is that patients say cannabis is helping with their pain. And pain is a very subjective experience. It's a very personal experience. And so if a patient is saying that, then that's certainly something that deserves our attention. Some of the more sort of preclinical studies are less clear on the analgesic effects of cannabis. But these studies found that oral THC was comparable to codeine. So it's a pretty small dose, 10 milligrams of oral THC. That's like a standard dose according to the government, and that's equivalent to that dose of codeine. And this is just further to what I said recently, that it distracts me from the pain and dwelling on negative thoughts. So the actual, it may be having less of an effect on the actual site of the tumor, or, the pain there, but you're not dwelling on it. You're distracted from it. Maybe you're more relaxed. You're not as wound up about it. And so if people are saying that they're using cannabis for, appetite stimulation, or they're using it for pain, but they're getting these other sort of side effects, then we may see a bit of a substitution effect because they may not need to use the same dose of their other medications. And that's what research from our lab has shown, that medical cannabis users were substituting cannabis for about 30% of them for their opioids, about 15% for their benzodiazepines, and just over 10% antidepressants. So just want to end off with a few slides on practical considerations. So when it comes to product choice, there's a lot of products out there, which is exciting, and it's great. Legalization has certainly kind of opened that up for us. And just a couple days ago, the government introduced now the second wave of legalization. So edibles and things like that are now going to be available. And so you have to decide what type of effects you want to get, how you want to use your cannabis. So there's a lot of prescription products that are available. Of course, there's standardization. Maybe your insurance company covers it. Maybe your doctor's more comfortable with it. And research has kind of shown that, at least with dronabinol and smoked cannabis, there isn't a huge difference in efficacy. So I think that could be a patient decision for sure. But when it comes to whole plant cannabis, there's something called an entourage effect. So as I mentioned, those terpenes earlier, and then all those different cannabinoids, those work together to kind of act synergistically. So that might be preferred than these isolated compounds. Sure, we know THC is effective for certain things, CBD for certain things, but perhaps there's other benefits that someone could derive by taking in whole plant cannabis. The route of administration is really important. As Pippa mentioned, smoking, vaporization, that isn't necessarily something that we want to be encouraging. But if a patient is, needing to use it for sort of like breakthrough pain or, just sort of a little tiny bit before bed, then you're able to much easier titrate by vaporization. But the oral, if you take it orally, it's going to last for way longer. So it depends what you're using it for. If you're using it for pain or for sleep before bed, then you may want to take it orally so that it sort of stays in your system for longer. I wanted to bring up the patient journal. So as Pippa said, some patients are maybe encountering physicians who aren't interested in working with medical cannabis, which would be such a disappointing experience when you're already going through that treatment. But if that is the case, or if, as a physician, if you're less sort of educated on it, you can individualize treatment with cannabis for a cancer patient or for yourself by using a patient journal. So you can start to figure out which types of cannabis work good for you, which types of routes of administration work for you, for what effects, and really start to learn what you need to be using. Because as I have mentioned a few times, cannabis is a name, but it is so many different drugs. And then finally, just to reiterate, start low, go slow. It's very important. The attainment of euphoric effects isn't required to attain symptom control. So if that's something that, you can start with very, very small amounts of cannabis. There's evidence of people microdosing cannabis and still deriving a benefit. So I think going for the lowest effective dose is better than just sort of like smoking all the joints. So that's what I would say. And then, of course, I wouldn't want to do, as a clinical psychologist in training, do this presentation without mentioning that it is important to assess for familial history of psychotic disorders, because of course there's that little bit of a chance of an increased risk of exacerbating those underlying pathologies. So cannabis is a multimodal treatment that can reduce the polypharmacy burden, and individualized treatment is necessary. I believe that ultimately cannabis can be an important medicine to relieve suffering during cancer treatment. Thank you.
That was fabulous. Thank you very much. Now we're going to switch gears a little bit. And I'm going to call on my colleagues, Dr. Bernie Garrett and Dr. Tarnia Taverner, to come forward and present some things, I think, on virtual reality. Yes. Yeah, virtual reality, so stay tuned.
Okay, hopefully you can hear me okay. We're going to talk to you a little bit about some projects we've been doing over the last few years using virtual reality for chronic cancer pain. And really wanted to just sort of describe a little bit about, is that not working? There we go. The history of that. We've got a team at UBC, myself, Dr. Taverner, and just to say we've got some research assistants as well, giving them a shout out. Who help us with this project. So, what is virtual reality and why is it something that's become sort of so prevalent at the moment? If anybody of you have kids who game, you probably have heard of it, you may have tried it yourself. It's actually become quite a sort of pervasive technology at the moment, particularly in the recreational area. But why has it become important now? Well, let's just talk about a little bit about what it is, and then we can talk about that. Basically, virtual reality involves using a head-mounted display and information technology to display sort of sight and sound and reality of an environment in 3 dimensions that actually moves with your head just as if you were in another place. So we use headsets like this one. This is an example of a recent headset which is completely self-contained that projects stereoscopically in both eyes, so you can actually see the experience of being in a different environment. If you hear birds sort of calling in the environment and you look around, you'll see them in the direction. And if you look around the room, you'll see exactly, you know, what you'd expect to see if you were actually in a real room. It's a bit difficult to explain. Out of interest, how many of you tried VR here? Quite a few, yeah. So about half of you. So
you
there's
get
a video here.
It won't be sound, but do some movement.
Now this is Gordon. He's got a headset on. You can see he's got hand controllers as well, because the modern iterations allow you to see your hands and move around. What you see on the screen above him is actually what he's seeing in his headset. And you can see as he moves his head, the screen moves as well. That's an environment where he's on an island.
Oh, look over in the other direction towards Thea's voice.
Yeah, and he's looking around this environment and seeing trees. There's a butterfly flying on there as well. And basically, as he moves around in his head, he moves around in the environment. It's quite sort of an interesting difference in the VR experience from looking at a screen. Because in a screen, of course, you're looking at something in 2 dimensions. Whereas in a 3-dimensional VR experience, you actually feel present in the actual screen, in the environment. Let's see if this is going to go. The batteries might be going on this. No. There we go. No.
Gone back.
Okay, so over to you, Tanya.
I'm going to discuss a little bit about the background to all of this work, which of course is pain, because we've been using virtual reality to, or trying to use virtual reality for the management of chronic pain, or to assist in the management of chronic pain. I think it's really relevant today because, of course, the opioid crisis, and we are having to look at different modalities to help patients in managing their pain. So pain is a significant issue in Canada, actually, all over the world, affecting millions of Canadians, and 1 in 5 Canadians will suffer or are likely to suffer with persistent pain for varying reasons. And this is always a striking statement, but chronic pain costs Canadian society more than HIV, AIDS, cancer, and heart disease combined. And often people don't actually understand, or we don't realise the impact of pain on society. But I know all of you will know somebody, if not yourselves, who have chronic pain. The impact on the individual is significant. Okay, it worked. So chronic pain associated with a cancer diagnosis is real, and it can be associated with the actual disease itself or because of the treatment. And what we're finding is the more where we're getting more cancer survivors, we're also getting an increase in cancer survivors with a chronic pain condition. And that chronic pain condition is hugely significant on that individual's life and on how they then recover, and then on how they then experience their life after their recovery. In many cases, as I've said, the pain is caused by the disease, but also by the treatments, and these can be treatments such as surgery, chemotherapy, or radiotherapy. The VR environments are hypothesized to manage pain via non-pharmacological attentional and distractive mechanisms. So it's quite complicated, and there is a lot that we don't actually understand and know, so it's really experimental. There have been several theories suggested to explain the phenomenon. And the most recent, and I think the most relevant, and the one that makes most sense to me, is the neuromatrix theory of pain. The neuromatrix theory of pain was developed by, as a follow-on from the gate control theory of pain, and you may have heard of those. And the neuromatrix theory is interesting and relevant because it's actually everything that we are, that we're born with, but also that we experience, will impact and inform how we experience our pain. Pain is entirely individual, and no one person experiences the pain in the same way. We experience it in a completely different way, and therefore we require pain management and pain therapies to be almost designed for the individual. We need to use multimodal approach. And VR can be one of those approaches. Oh, I don't know why I did that. Anyway, we believe that, or we think that the VR therapy may work by dampening the acuity of the pain modulation system by acting on the signaling pathways of the neuromatrix and therefore producing analgesia, but also reducing some of the pain mechanisms. So I'm going to hand you back over. Oh, actually, no, I'm going to continue on. Yeah. There are new opportunities. Since 2012, the virtual reality has become much more affordable. I know that when Benny and I started this work, not only was the equipment quite expensive, it was also very cumbersome. The new equipment is becoming readily available, and it's much more practical. And this then allows us to be able to use it more in research, but also makes it accessible for more people. Before we did our first study, we did a pain research, or VR pain research review, and we found that most of the studies that had looked at the use of VR for pain had actually used it for procedural type pain and for acute pain. There was nothing around its use for chronic pain. So that kind of made us more interested in wanting to investigate this and to see if we could actually see if the use of virtual reality would have some sort of effect. With the review, we found that there was moderate evidence for short-term effects on physical function, but little evidence existed for any longer-term benefits. That's not working. Nope. I don't know what it is. Oh, okay. Our first study was a pilot case series. We, in 2015, it seemed like much longer ago, but 2015, we tested the use of virtual reality on 10 adult patients, and they had chronic pain conditions associated with varying different causes and backgrounds. And for 1 month, each patient used a VR therapy for 40 minutes every other day. Pre- and post-pain levels were recorded using the numerical rating scale for pain. And I just want to acknowledge that actually using a pain intensity scale isn't that useful for chronic pain, because pain intensity isn't really what a chronic pain patient focuses on the most. It's actually their impact on their quality of life, whether they can sleep, whether they can continue working. So we also did weekly neuropathic pain rating scores as well, because we wanted to see if there was an impact on any of the neuropathic pain experiences. And our findings were that there was some evidence for immediate and short-term pain reduction. It was very varied though, and again the numbers were very small. The applications that seemed to work were very varied as well. But that doesn't surprise me in that people experience their pain in very differing ways, so therefore their response is going to be varied. And some evidence was found for short-term effects on physical function. We didn't find any evidence to demonstrate that there were any longer-term benefits. I'm going to hand you back over to Bernie.
Wait for the next slide. So basically that was like a pilot study. We were interested in seeing, did it have any application in a chronic pain population? So since 2014, there's actually been a lot more research in this area. It's sort of exponentially increased if you look at the publications now. And there's a lot of pain studies. They're mainly in acute pain and procedural type things such as dressing changes, cystoscopy, a whole range of things. But there are some chronic pain ones now arising. Spinal cord injury, for example. Phantom limb pain is a popular area where this has been used. So we were interested in seeing would it have any application in cancer pain. We'll wait for the next slide to come up. And I seem to have gone one back there. Did we get one slide missing? Yes, thank you. Our current trial, we've got funding for a trial which we're currently undertaking, an RCT, looking at 100 patients through, using cancer patients with chronic pain, comparing our VR therapy to multimedia therapy, which is basically the same environments but in a 2D laptop environment for the control. Because we're interested in what is it about VR that actually sort of seems to work for pain control. So we've got two treatment conditions that we're using. One is introspective, a meditative relaxation type environment. That seems to work for some people, they like that. Another one is cognitive distraction. So some people don't seem to like the meditative approach to relaxing, the introspective sort of environments. They prefer to be distracted with something that involves problem solving. So cognitive distraction is used for that. Let's wait for the next slide to come up. We've basically got sort of some moderate results already, but this is ongoing. And if you can see these are the types of environments we're using. That's an example of the one that's the relaxing environment. They're on an island. It's very peaceful. There's an eagle flying around that they follow, and there's spots where they take guided meditation during the 40 minutes they're in the VR. There's also another one which involves them solving puzzles with light beams in 3D, and that environment is much more engaging about figuring out 3D manipulation, so it requires a bit of thinking to resolve that. We have some practical issues we've discovered. There are users' hardware limitations. People, for example, you still need quite a powerful computer to run VR for a lot of things, although headsets like this, which are self-contained, are now coming available. Also, people's technical skills. Obviously, older patients are sometimes a little bit more reluctant to engage with newer technologies in terms of complex information technologies, and some of these are a little complex, although most of them adapt very well to it once we've gone through the familiarization process. But of course, with chronic patients, you have all these issues of unplanned illness, surgery, dropout, and things like that affect their ability to follow protocols with an RCT. Also, personal preferences regarding design have come to play, and we've put a paper together on that because a lot of the applications for VR aren't designed for pain patients. They're designed for recreational games or relaxation. And lastly, interesting ones, there's a bit of evidence as well about tolerance to what we call cyber sickness, and this is the motion sickness that you can get by using a VR headset, which occurs simply because there's a disconnect to what your body's doing and what your eyes seeing. Your inner ears might be telling you that you're sitting in a chair, but your eyes are telling you you're flying through an environment. So there's a few things like that to be worked out. So our ongoing work still involves the RCT. We're still recruiting. If you have patients who might benefit from this, the beauty of it is we can give you the whole setup to the patient to work in their own homes. They can take it home with them, use it at home every day. So we're still recruiting for that. If there's patients' representatives who want to talk to us, we'd be happy to talk to them to get them set up. We've also just done a focus group study, which has proved some interesting results as well with patients who've been using this. And at the moment, we're doing some EEG studies with the meditation VR work as well. You can see a picture there where we've recorded EEGs of patients undertaking VR experiences to see if there's any effects on changes in the brain waves, and we're analysing that currently. So here's some quotes just to finish up. You can see there some patients really do benefit from this quite remarkably, and for the period they're using the VR, actually virtually manage to eliminate their pain or forget about it. It doesn't last any time afterwards, it seems, but you see some people see that it is quite effective. Also, some people don't find it effective. It's very individualized, so it's a personal thing. But we are finding some quite powerful results with some patients with it. And really that pretty much wraps us up. In the future, just things that we're looking at. The quality of evidence and value of VR as a clinical sort of pain tool is still very immature. We still need a lot of work to do to show it works, how it works, and one size doesn't fit all. That's something we've learned quite early, that it's very important. It's not just the VR, it's the environment that the patient's in, and it's got to be adapted to meet their needs. For example, we had one lady who had a fear of water, so the environment was around a lake. She didn't like that at all. So these personal things are very important to establish. Adverse effects is important. Cyber sickness, another thing is people get some eye strain from it. You can only use it for about 30 or 40 minutes because you're actually focusing right in front of your eyes on a headset, and after about 40 minutes your eyes get a bit tired with that. The headsets are getting lighter, so we're getting less other physical problems. But there are things that we need to think of as well. Thank you very much.
Up next? I'm up next, right?
Yes, you're up next.
Okay, you said medical assistance in dying. Which slides are coming?
Which ones are you?
I'm doing NavCare.
Oh, sorry. Right, sorry, Barb's talking about NavCare. She also does medical assistance in dying.
I do.
So I got mixed up.
That's okay. No, it's true. I have another program of research. Yeah, I don't know about that. We'll see. We'll see. Thanks, Joan. Yeah, okay, I'm going to talk a little bit about NavCare, and just let me start with a case scenario. So you have an advanced cancer patient. He's 74 years old, he's living at home, he's taking care of his wife of 40 years who has dementia. He has a fall at home, he must be hospitalized, and of course his wife has to go into hospital with him. She ends up passing away while in hospital. He returns home, he's bereaved, he has decreased mobility, and his primary meaning in life is now altered. So the interesting thing now is this patient is going to go on one of two trajectories, right? He's either going to go on a trajectory of increasing frailty, which will lead to a fairly rapid death, or we're going to interrupt that at some level and help him to renew his quality of life and his meaning in life. And that's what NavCare is about, making connections, making a difference. So the goal of the NavCare program is to improve the quality of life of adults living at home with illness by providing specially trained volunteers to create connections to community and to provide caring, consistent emotional support. So I like to acknowledge my colleagues. I work on this with Wendy Duggleby at the University of Alberta. We've had wonderful funding, including the Cancer Society. I have a lovely investigative team really from across Canada, so I like to mention them. And probably most important, the partners that have gone on the journey with us. Because this has been a long one. This has been a 12-year journey. It started with ethnographic studies in rural palliative care, trying to decide what seniors needed best. We did some pilots. We just finished some knowledge translation studies from sites across Canada, and we're now scaling it out to try and build the evidence further to do a policy push. So who does NavCare serve? As I'm sure you've seen this, that 1 in 5 seniors report that they experience emotional distress and have difficulty coping day to day. So what are some of the, I'll miss this slide, I'm going to make up a little bit of time, so forgive me. These are some of the quality of life issues that seniors are struggling with, that NavCare seeks to address: loneliness, recent loss, mobility or sensory challenges, multiple concurrent life changes, increasing disengagement for sure, coping with multiple decisions, and difficulty finding access and resources. Over this 12-year journey, these are just some of the things that we've learned that specially trained volunteers can do in making a difference in quality of life for these seniors. And what's really interesting is all of these concerns directly impact health and healthcare, and yet many of them don't fall within the responsibility of healthcare providers. So it's trying to fill in that gap. So when you look from a policy perspective, our goal is really to include okay, why are you doing that? It's really to Increased quality of life, which is very different than cancer navigators, right? These volunteers are not navigating the healthcare system. They're navigating quality of life issues. We're trying to capitalize on all of the baby boomers who are retiring, needing good volunteer roles, optimize available resources, and build social capital, and of course a palliative approach, right? Providing early support. Right in keeping with the compassionate community movement, it actually is a mechanism for communities that want to do compassionate communities, and it's a program that they can mount to do that. So here is the NavCare intervention in a nutshell. The volunteers visit clients in the home. They usually have a 2-to-1 ratio. They get evidence-based education in a 2-day workshop, ongoing mentorship by a nurse navigator. It's very community-based, so it's adapted to that context. It rests on community champions. And then we have a robust evaluation across the programs. So these, we have now a toolkit created. These are the resources that are available to any society that wants to start NavCare. So who implements it? It's largely hospice societies. Just as we have bereavement volunteers, hospice volunteers, we now have upstream volunteers called NavCare volunteers. And they do follow clients right through until death. We've done a few trials across Canada with non-hospice societies, most notably with the Curdy Centre in Calgary that serves thousands of seniors in downtown Calgary, and they layered this on their home visiting program. We now have actually 16 sites have finished the research, 8 were sustainable after the research period without further funds, and 11 are in various stages of development right now. So we have really all the way from Vancouver Island to Newfoundland. So what is the evaluation shown in a nutshell? Volunteers are incredibly satisfied with the role. They're really resourceful in providing navigation support. And we, of course, we rate their self-efficacy in navigation along our 36 some odd competencies. And they've had good self-perceived efficacy. Clients feel the service is very important to their care. They typically rate it 8 to 10 on a scale of 10. Very tangible benefits transform their experiences of living with a serious illness, the vast majority of which are cancer. Stakeholders feel it's meeting an important need, it's well designed, but key supports are required for sustainability. So I've just actually put a couple of quotes in here that you can read that are fairly typical. We have a whole thing of qualitative evaluation about what seniors say, but certainly one is a safety net. As this fellow said, I need something that's reliable in my life. The navigator is usually a minute early because when you're in this condition, you really need stability and security, knowing that this is the one thing you can hang on to when things go sideways. It really has been profound. I'm getting all weepy as I think about this. Another thing, very much in terms of decision making, as you know, seniors, particularly those undergoing cancer treatment, have so many decisions to make. And so they talked about how great it was to have that person who wasn't being affected like a family member, through which they could talk through their decisions. Definitely transition support. These volunteers were helping them with signposting. This is what you can expect to come next, and this is perhaps what we need to be thinking about. And certainly engagement. This was actually, we were quite surprised at how profound re-engaging was in this intervention. The fact that people who had really started to stay home and withdraw from everything, including their physician visits, were once again getting remobilized, doing activities again, and paying attention. And I love this last quote. It's focused on living the best you can with whatever you've got each day of the year. That's been a real bonus for me, and it's led to other connections. Certainly some element of planning has been part of this, and particularly helping seniors do planning that costs something like wills. So helping them to find how to do that free. Our implementation lessons, client recruitment has been the most challenging. So connecting the volunteers to the people in the community, because remember, these seniors are almost always being seen through the primary care network. They're not on the palliative system yet, they're upstream. But communities that have great champions, have got a lot of organizational community capital, good volunteer coordinator, they've just done really, really well. Some of the challenges have been unclear messaging, massive organizational change, part of which was related to the MAID initiative for these hospice societies, which you'll hear about next, and inadequate support of the volunteers because you're asking them to take on quite an intensive role now. And that's for me. Thank you very much.
going to have a presentation on medical assistance in dying, and I'm pleased to welcome Dr. Harish Handel. So welcome. And this is our last presentation, so jot down those questions and comments. I think it's on.
Thank you very much, Harish Handel, H-U-N-D-A-L. I'm going to tell you a story. I'm going to talk about Alice's great demise. This is Alice. This is about 20 minutes before she received medical assistance in dying. And I'd like to thank her daughter, Carrie, who is willing to share this story. So Alice was an individual who reached a point in her life where she's no longer able to lie down. This is the best position for Alice. She's actually stayed in this position all day because her muscles that helped her breathe had reached the point of weakness that when she lay down, she stopped breathing and started gasping for air. As well, in terms of being able to do her activities of daily living, she was completely dependent on others. She could not even scratch her nose. And for the few months before she died, she'd been having episodes where the food that was in her stomach would go back up and down into her lungs, causing burning and inflammation. And that was her fear of how she would die. So Alice was a very incredibly bright individual. And Alice spent hours listening to the CBC when the medical assistance in dying, the bill was going through Parliament and all the different things. And she put a lot of thoughts contemplating about how she wanted to die. And so the patient actually does the heavy lifting. The patient, what I tell individuals is that when someone asks about medical assistance in dying, it's actually an invitation to be invited into a conversation about what is a good death. What is suffering to you? What are your expectations? And what do you know about your condition? And what are your options? So Alice had these conversations with her family physician. And the family physician worked through on his own regarding how did I feel about this. So a physician can say, you know, I'm really uncomfortable with this. But as a physician, your role is, or as a nurse practitioner, your role is to say, if I'm uncomfortable with this, I still have an obligation to provide comfort and care to my patient. And I also have an obligation to say to my patient, I'm not comfortable, but I know where you can get the information that you need and the support you need through your journey. So Alice went around preparing everyone, including her family physician, multiple conversations. The determination was determined by two physicians. So her family physician at the time said, right, I've reached a point, I've cared for Alice for so long, I feel an obligation on my part to be involved in this. And she contacted through her daughter the Medical Assistance in Dying Coordination Center. So as part of the legislation, each of the health authorities has a coordination center. And so she received information, she had to initiate the request, had to have two people who weren't going to benefit from her will. Alice had been quite clever. She got rid of all her belongings. She was able to call family members who came from all over North America to visit her, and she got to say goodbye over time and said, you can have this painting, you can have this, and you can have that. So over a 3-month period, she prepared the people around her and her caregivers. She was in a long-term care facility. So it's the Good Samaritan Society. And Alice spent time preparing the staff who cared for her every day. And when she contacted the coordination center, individuals went in and we had conversations with the staff about their feelings, about what does it mean to die, what's available, what is the process. And so a lot of groundwork, a lot of preparation. Is that the right one? So there was meeting with administrators and staff, and in fact it reached a point when after the second assessor, so there has to be 2 assessors who have no conflict of interest, individually assessing whether A, this person is capable. And capability is not about finance, it's about, do you understand your medical condition? Do you understand the nature of your illness and what options are available to you? As well as, do you know what you're asking? You're asking for someone to help you die at this point. The caregivers gradually over time have been brought along, the patients and brought along her family. And that was the most interesting part. As you can see, her daughters are smiling. And at first, when the daughters go, when Mum first mentioned to this, I was just shocked. You know, and my first reaction was, I don't want you to go. I don't want you to go. And then it opened up a conversation about loss and grief. And what patients have described is that they're helping their family members move through the grief process before they die. And that's not happening at the point of a 3 o'clock in the morning phone call that Mom is gone. It's happening a week, hopefully at the latest, but much earlier than that in that patient's journey. And there was respectful agreement between the staff and in terms of who would be there, who would provide what role in the process. So setting the table, Alice set the table. She did most of the conversations. We essentially supported her in having those conversations and supporting the staff and the providers. In terms of the actual day, Alice prepared for it. She had her favorite pictures out, etc. So people often ask, where can I have medical assistance in dying? Most medical assistance dying is actually has been provided at home for individuals with a family. We've provided medical assistance dying in every setting in the hospital, from surgical floors to ambulatory care to the intensive care unit. It's been provided as well in long-term care. I have to get used to using long-term care, given the last name was culturally inappropriate, as well as the most interesting examples is someone who had medical assistance dying provided in the outdoors around a lake, just surrounded by family. Who can be present? That's up to the individual, and to have that conversation and prepare people for the expectations. So Alice had her daughters present. One person has had his 3 ex-wives present. It's variable. Where you cannot have this, one person actually requested, can I have medical assistance dying while I'm watching my favorite hockey team? Actually, no, it has to be appropriate. There's a certain degree of decorum. And the family needs to, the individuals present need to understand this is still, the choices involve either, most often is a series of injections, right, through an intravenous. There's 7 injections in a row. People often ask, it's the exact same medications you would use when you go in for surgery, except at far higher dosages, right? So what people are told at first is they'll simply drift off to sleep, then we will provide the anesthesia, then we will stop the diaphragm from breathing, and it'll be about 7 to 8 minutes, the entire course of the intravenous approach. There is an oral solution as well. That can take a bit longer, but you still need the intravenous as backup in case the person throws up or the person's simply not able to tolerate, for whatever reason, the oral solution. After people who've been at these events, the feedback from the providers. One individual is an obstetrician gynecologist who does a lot of medical oncology with women with ovarian cancer, is the case that has primarily been involved in. And he said he approached it with trepidation at first, and then afterwards his reflections were, it was much like being in the delivery room to him. He said there's anxiety as the day comes. You're wondering, you don't want to do anything that ruins this for the family. You're overly cautious to a degree that you never are in any other procedure in medicine. He said even his hand shook a little bit in terms of the degree of tension and anxiety this elicited. Then afterwards, he's completely surprised by the family hugging him. And this is what the providers have said, is that they feel privileged to be in the room with that family, to be present during this, during the provision process. And the families afterwards, it's the reaction for the people present is usually, there's an interesting sense of joy at the passing and being present. One person asked, well, how do people dress for this event? And yes, they do have parties, some of them, it's going away parties. One woman wore her wedding dress, lay on the bed with her bouquet that she had from 30 years before, and her husband curled up next to her. And that was their goodbye. Intimate moments. And afterwards, the staff do debrief. And so there was a debriefing with Alice's family afterwards and the staff of the Good Samaritan Society. A letter was sent from the CEO of the Good Samaritan Society to the health minister at the time to say any individual in our care should think of our place as their home, and we will support them as choice. And in the end, it really is every individual's journey. We are not great at being able to say that your suffering is less than another person's suffering. And others may judge, but at the end, it's not their journey, it's yours. And I think that's the theme for this talk today. Here are some great ways of helping you with that journey, and of also opening up those conversations about death, dying, suffering, and the value of your life. Thank you.
Oh. Wonderful, thank you very much. Could I invite our panelists up and give you all just a few minutes to think about any questions or comments that you have? This has been a really great panel, because what we've been offered is really a lot of choices for patients, but what I saw throughout was this opportunity for patients and healthcare providers to really personalize the cancer journey for patients in ways that will support symptom management and also quality of life. So I'd now like to invite some questions. Yes?
I've been a nurse for a long time, and sometimes reality sucks. Frankly, it's cruel. And I see that you all offer options for individuals to choose different realities. But I've also seen working in hospice, working in long-term care, working in acute care, sometimes people dying make choices for themselves, but sometimes they make choices for their family. And that's okay. But what do you do about when there's individuals that are maybe preventing them from choosing their reality? Because I see that sometimes happens, that a key family member or a key group or a key system prevents that. I don't know if anyone wants to comment on that.
Yes, can I invite anyone to take a stab at that? And they can be more than one, obviously.
I could maybe have a stab at that. I'm thinking of situations like if somebody has accepted that they are not going to get better and they would choose not to have more dialysis, more chemotherapy, decide not to have a transplant, and then other family members in their anticipatory grief are not able to let them go. We do see this not infrequently, and it's often the way to solve that is to have people sit down together. I can think of one particular story about a family where we had an elderly gentleman, English as a second language, and his family had told us before his appointment at the Pain and Symptom Management Palliative Care Clinic in Vancouver, don't tell him he has cancer. Like, it'll destroy him. And so, I mean, this man clearly knew he had cancer. We talked about it a lot when the family weren't there. But it ended up being very difficult. So we got the family together, and then in front of the family, I said to the patient, "Please, can you tell me what you understand about your illness?" And he said, "Well, I have cancer, and I think it's really a shame that my family won't let me talk about it in front of them." And so that problem was immediately solved. So I know that's not always that easy, but trying to get them all on the same page by talking rather than skirting around the elephant in the room tends to be the most successful.
That's great, thank you. Yes.
I probably could have questions of all of you. Thank you for today. Dr. Hundal, I have had a close friend pass by assisted death or assisted passing. I still personally struggle with it, mainly because they were the first person I know close to pass in this way. Has there been any thought put around those who are left behind, who may still, like I'm more than a year later, and I still, it was the worst feeling to know on Tuesday I will pass Saturday at 6, and the days count, and the hours count, and the minutes count, and it's almost, although you're grieving, when you're sitting in your home, knowing this now is your reality, and I'm just wondering if there's been any thought to that after piece for supporters.
So in terms of at Interior Health, we do have a chaplaincy and pastoral care. As well, we do encourage people to access social work support, etc., talk to their family, doctor, in our First Nations communities as well, to have those conversations. Sometimes it's very hard for people who haven't processed what's happened, right? And sometimes we do get into the situation where the person has decided that this person, a family member or friend, they don't want them present. They may not even tell them that this is happening because of the fear of the reaction. And then the friend wonders, why didn't they, right? We live in a society where individual choice is valued. So I do encourage you to reach out. There are supports out there through your family physician or nurse practitioner or your community to have those conversations and sort of process that grief that you are feeling.
And Barb.
I can probably weigh in on that a bit as well. There's actually some really interesting evidence now coming out that the bereavement trajectory is actually quite different for families after a MAID death. And so the Canadian Association of MAID Assessors and Providers, on their website, I believe they list family support services. They've actually started several new organizations within Canada that acknowledge that this journey might be a little bit different.
And it can help to connect with other people who have been there, been through the process, to be able to reflect on what was their experience. Because we actually find, and I think this speaks to the NAV care, that having social support is very protective for individuals, regardless of whether it is cancer or mental illness or whichever condition. And often when you yourself don't have those supports, it's very difficult to cope.
Just a little plus thing as well. When it first came out, when it became legal, it happened actually quite precipitously, and the healthcare system was frankly not ready. And a number of the procedures done at the beginning, I think there was a lot to be desired from the process, but I think we're getting better at it now. And even in the last year, I think there's significantly more awareness of the need to prepare for these things and address them in advance. So it's not as difficult now as it was even over a year ago.
The great thing I think happening now is that we're actually studying people. Like most at the beginning was reacting to the legislation coming in. As I can tell you, at the time people even didn't know what the legislation was until about 3 days after it had been passed, even though the previous government had over a year to work things out, and
There was a big rush, wasn't there?
it's a totally big rush. And I think it's an evolving conversation, but one of the things that people talk about is dementia and its capabilities. Can't I make that choice earlier, before I become demented? Why do I have to be totally aware at the time? And that's a very interesting conversation and how that will play out. And in terms of suffering, we've sometimes had how do you measure it? Like, how do you know when a pedophile calls the aid center and says, I would like to have medical assistance in dying because I know that I am going to offend, and that I regret what I do. Can I? And you say, well, no, you're not eligible at this time. And we have to, as physicians, and sometimes we get quite criticized for this, we have to follow the law and the rules in place. And this is probably the most stringent and controlled, and rightly so, service or support that we provide to individuals.
Great, thank you. Yes.
I just wanted to follow up on that, of your comments, and the question and your comments, because certainly before the law changed, there has been some really good books about respect for your decision in dying, and that. Is it too new for books to have been written about the journey from, I'm really curious, from the bereavement trajectory being different as well. Is there anything been written yet that would help people that are going through the process that you know of?
So Carrie Taylor has done several articles. You can look up Carrie. She has, and there's from the National Society, National Group, there are contacts and information. I think it's evolving, right? And so new, and people at the initially, remember it's only 2 years, really, 2016 or so. And that within this is the whole idea of simply, let's get the process as right as we can, is where the focus is. And now we're finally having the research arm of care catch up to it, and being able to, and we're also embarked on studies of actually following the providers and the assessors, and what is the impact on this, on these individuals over time, and what is an appropriate time between provisions, you know, because they process as well the situation and need a recovery time to come back. To expect someone to go back to back as if it's a tonsillectomy is really inappropriate, and if anybody did that, I would be quite shocked myself. That if you're a human being, this comes from a place of compassion, and it's emotionally draining.
Great, thank you. Yes.
Sorry, another question on virtual reality. Has there been any thought or process around patients with chronic pain who have epilepsy? Are they eligible to participate in something like this, or is it like, have you investigated that, or is it just off the table?
It's a good question, and when the technology first came out, the first thing you saw when you put on the headset was a warning to do not use if you have any of these conditions, including epilepsy. But it's actually not been found to be a particularly problematic or likely to stimulate epilepsy. Obviously, there are some environments with bright flashing lights and things like that that could trigger people, but the majority of them aren't, and it's pretty safe. Also, they didn't use it with children to start with because they were worried about the effects on them. So I haven't heard of any studies being done particularly for people with pain in epilepsy. Our studies are focused on chronic cancer pain, which is a variety. And in our pilot series, we had people with a variety of pain, including migraine, quite severe recurrent migraine, and various others. And they used it without any problems. So it's certainly an area where the research could proceed with that group of patients. But as I said, I think it's in the very early days at the moment, and there's very limited applications. So where it would be useful and what sort of applications would be most useful for those patients sort of remain to be seen.
That's great, thank you.
Yes.
I had a question for the speakers talked about cannabis.
Great, go ahead.
One of you had mentioned that there's a potential interaction between CBD and immunotherapies and the immune system. And that's news to me, not something I'd heard before. And I was wondering if you could expand on that, but possibly more broadly talk about some of the other harms that might be there, either in the literature and known or theoretical, between cancer treatment, and cannabis and its derivatives.
Shall I attack the immunotherapy one briefly? Yeah, so there's only been, I believe, one study, and it was a very small study, and it was retrospective, and it's not something that you would hang your hat on, but it did suggest that in a group of patients who were receiving nivolumab, which requires the body's immune system to work to try and kill the cancer cells, people that were using medical cannabis or using cannabis for any reason seemed to have a lower response rate to the immunotherapy and a more rapid recurrence or relapse in those that had responded. But it didn't make any difference to the actual hard outcomes, so progression-free survival or actual survival were not affected by the absence or presence of cannabis. Plus, the cannabis was not explored in any great detail. So I think there is a theoretical possibility that high-dose CBD may well have an immunosuppressant effect and probably shouldn't be combined with immunotherapy until we know more. But at this point, you know, if someone's using something that's low in CBD and they're finding it tremendously helpful for their symptoms, you know, as long as the immunotherapy seems to continue to be working, then I probably wouldn't make a big deal out of it. And at the end of the day, even with these immunotherapies, I know there's a huge amount of song and dance about them at the moment, but in actual fact, for some of them, they're just prolonging the agony, to be honest. People are getting a few months out of it. Great when you get somebody that has a few years, but most of the time you get a few extra months at quite significant costs, both financially and in symptoms and time spent having to have medical appointments. So they're not, you know, they're not the best things to slice bread. Sorry, I've mixed way too many metaphors there.
Do you want to add anything, Michelle?
I mean, the only other thing is kind of the theoretical risks of using cannabis for cancer. I'm definitely not a cancer expert by any means. But I think that the only other thing I've seen is again related to CBD and some concerns about CBD and the liver. So I think that, you know, just using moderate doses and like I said before, just that lowest effective dose, I think you're going to mitigate most of the concerns around cannabis.
Oh, we have time for another question. One more. Any last questions? Yes. Go ahead.
I have one more.
Good. Oh, this is fabulous. Go ahead.
The cannabis group. Recently in the news, we are hearing about the government in British Columbia being so much slower than other areas. I know people that have had to travel from even the lower mainland to Kamloops originally because they were the only one open. Is there any thoughts around that, especially like rural communities where people yet do not have access? Is there any pressure being put on the government to speed this process up, or can you comment at all on that?
Comment on the storefront dispensaries are meant to be purely for recreational purposes. They are not to be used for medical use. Though many patients are, they won't get proper medical advice from them or can't be relied on to get good medical advice from them. So they should always use the online licensed producers, which are available to, as long as you've got a street address, even if you don't, you can have it delivered to a nurse's station in a remote First Nations reserve. You can have it delivered to the nurse's station. You can get it. You just have to use the official medical process, which has been there for some years and continues to be there. So there's no excuse for anybody to not be able to access that if they have sufficient help to negotiate the actual process. You have to be able to sit down and work a computer and fill in a registration form and get somebody to sign your medical authorization for you.
So that can still be quite a barrier for a lot of people.
It's a barrier, but at least it's a process. It's not perfect by any stretch, but it is available to every single person in Canada.
Do you want to add something?
Yeah, the only other thing I'll add is that there's just a new study out of Colorado, and they were at a recreational cannabis dispensary. So they had 1,000 patients, or 1,000 just regular adults surveyed, and 70% of them were using cannabis for a self-identified medical reason. So I think that if you do have barriers, I think that there is, unfortunately, for kind of the relationship with maybe your physician, but there is still an option for patients to access cannabis through the mail order system that the government has set up. So if you live up north and you don't have a storefront dispensary and you can't get authorized by your physician, you can still order through like BC cannabis stores.
Great, thank you. Well, this has been a really, really interesting session, and our time is up, and we have a reception out there. But before we go, I want you to join me in thanking this panel and thank you for all your great questions and comments. And I invite you to go out into the lobby and join our reception. Thank you.
I know, I was curious because